Bismillahirrahmanirrahim. Bismillahirrahmanirrahim. Assalamu alaikum, ladies and gentlemen. I request you all to please rise for 1 minute silence, followed by the national anthems of the two countries, Pakistan and Turkey. Ladies and gentlemen, I'm Hazima Mozam, Program Manager from COMSTECH. I welcome you all on behalf of COMSTECH, COMCEC, and Middle East Technical University to the capacity building events, which includes 2-day training program and 1-day workshop. These activities are funded by COMSTECH in collaboration with, is funded by COMSEC in collaboration with COMSTECH and Me Too. We are very grateful to all our participants, distinguished guests, and speakers who are joining us here in person, and participants and speakers who would be joining us virtually. With this, I would request our esteemed distinguished guest, His Excellency, Muhammad Cyrus Sajjad Qazi, Ambassador of Pakistan to Turkey, to please come on stage, sir. His Excellency, Professor Dr. Muhammad Iqbal Chaudhry, Coordinator General COMSTECH, on stage, please. Professor Dr. Ismail Abdul Hamid, Director Programs and Project, Islamic Organization for Food Security, on the stage, please. Dean, Me2 University, Atuk Ospenic, on the stage, please. And the head of the programs from the COMSEC office, Mr. Mehmet Artikish, on the stage, please. Okay. Distinguished guest, we are extremely grateful for your time and coming and joining us here today. With this, I would request our two technical focal person for these events, Dr. Shahid Mansoor from Pakistan and Dr. Emre Aksoy from Turkey, for their opening remarks. Thank you very much, Mark, for joining us today. With this, ladies and gentlemen, I would request you to please welcome Professor Dr. Mark Tester. He's a full professor from KAUST University, King Abdullah University of Science and Technology. The title of his talk today is Future of Crop Breeding, New Technologies for Food and Nutritional Security. With this, I would request, as well as Professor Dr. Shahid Mansoor, to please introduce Professor Mark Tester and his domain of research to all the participants here and virtually.
Thank you, Hazima. I think it's an absolute pleasure to welcome our dear friend for many years. I won't say how many, but I think it's around 18 years that we have been in contact. Wow. Mark is a perfect human being and also a very good scientist. And he has been, despite his very busy schedule, he's whenever we call him, he responds to our requests. So I'm very pleased that he's, as Hazima has told, that he's a professor, but by training, Mark has done some wonderful work in plant physiology and also understanding how the plants respond to drought and salt stresses. Mark is also has done some of the very pioneering work on some of the genomic technologies, so to discover some of the important genes, as I was discussing about the orphan crops or the less emphasized crop, or even wild relatives of the crop. So he has done a lot of very interesting contribution in real science. By utilizing some of these wild relatives, especially in tomato and a lot of other crops. Mark is a very distinguished scientist. He works in Saudi Arabia, and one of the key aspect of his research is about how we can use saline water for irrigation and make this dry land very productive. Mark has some other engagement other than KAUST. I won't, so these are more into, generally looking into the science technology. And so Mark is a very good ambassador of science. So he has been able to push the science agenda in Saudi Arabia and OIC countries. With these remarks, I will invite our good friend, Mark Tester, to deliver his talk. Thank you very much. Yep. So it's all yours, Mark.
Thank you very much, Shahid. It's lovely to see you, and I'm very sorry not to be with you all in person. I think this is an excellent workshop. It's been a very long time in the oven to get to this stage. Of course, COVID has slowed us all up terribly. I don't know how many are actually live in the audience. Maybe somebody can tell me this. I can see there are 93 online, but is there a live audience as well here?
Yes, sir. We have around about, I guess, 45 people sitting in the room as well. And Mark, I would request you to please share your slides as well when you speak.
I absolutely shall. Yes, I'll do that as soon as I finish my little preamble, which is now. So it's welcome to everybody. And yes, as I say, I apologize again for not being able to be with you all in person. But it's an honor to be able to give an introductory talk today. I'm going to provide, and I asked this with Shahid, and my understanding was to provide context for your workshop. I'm not going to go into technical details on gene editing. I'm just going to try to give you some examples of what you might be able to do and try to remind you all to not just get into all of the technical details, which of course you must do, but don't just, in inverted commas, do the technical details, but remember why you're doing what you're doing and think very critically and analytically about the end result that you are wanting from all of your clever, skilled, technically sophisticated experiments and manipulations. So for me, I think now in particular globally about food security, and not just the amount of food, but also the quality of the food. I don't mean quality in that, oh, I must have a tomato that has got no blemishes. I'm talking about quality in terms of, well, the carbohydrate composition, of course, to provide the food that people want to eat with the texture and taste that people want to eat, but also with the right mineral and vitamin compositions. So hidden hunger is another massive issue and is of particular importance in South Asia and West Asia. So I really do urge you all to think extremely carefully about what you are doing and why you are doing it. So when we're talking about crop improvement, we want to be very careful to define our target. What does your country or your region really need? Not what you want to do, what is needed for your country or region. So Shahid and I were just talking before this presentation about drought and how to try to make land that's very dry more productive. And this is an extremely good sort of question. You can also think about, that's right, Shahid was thinking about fodder, because that can be a good thing to grow where you have low water supply, but it's also needed in the Pakistani context because you're needing to feed animals, because people need to, and there's an increasing need for meat production. I should say, actually, I'll talk about meat production at the very end of my talk, but it's an interesting global issue, actually. Once you've identified what's really needed, you need to identify how to address the target. I list here the full breadth. Identify the crops. In fact, you could even add in front of that, identify the systems that you're using. The systems, and by that I mean open field versus greenhouse, and irrigated versus dry land. The crops that are appropriate, the processes which are needing to be altered. If you've got the system and the crops defined, then what processes need to be altered? Do you need to make the plants more salt tolerant or drought tolerant? How do you need to make those drought tolerant plants? They need to have deeper roots, or do they need to have shallow roots? And don't get stuck by paradigms that are pushed upon you by old men. It's usually old men, all right? You don't want this. Question intelligently. Don't just question, but question intelligently the processes that you might need to be altering. And an example is, in fact, drought tolerance. From my experience living in southern Australia for some of my career, we have a very erratic rainfall. So you have drought where you have maybe 3 or 4 weeks with no rain, and then there'll be a light rainfall, and then another 3 or 4 weeks with no rain. And in a lot of southern Australia, we have very shallow duplex soils where at 50 centimeters down, 30 centimeters maybe even in some soils, there'll actually be a B horizon in the soil structure, which is toxic. And you actually don't want to have deep roots going down and then suffering toxicity, or even worse, translocating toxic ions from deep down up to the shoot to inhibit shoot performance. So, and because you have these light rains during the growing season, you actually want to have shallow roots to be able to harvest those rains when they do come during the growing season. So having a steep, deep root is not necessarily desired for a ability to grow with limiting water supply in all environments. So think very carefully about the processes that you need to be altering for your particular environment and need for your region. And then of course, once you've identified your systems, crops, processes, then you start to need to identify the genes. And don't just think about genes. It's the variant of the gene which is important very often, not just the presence or absence of the gene. So the alleles of the genes that you're needing to alter. People often think about just knocking out a gene or overexpressing a gene and then constitutively overexpressing a gene. This is often comes with more detrimental side effects than beneficial primary effects. And you need to have so often not just having more of a gene expressed, but often you need to have it in a particular cell type and have the expression targeted. So there's a lot of subtleties, and these subtleties are not just a pedantic academic just trying to show how clever they are. These can be often extremely functionally important, critically important for the appropriate operation of a particular gene, a particular variant of the gene, for a particular process to be manipulated to have an end result on the crop, the system, the yield. Then when you've done all of that, you need to identify the appropriate technologies to be using to achieve that particular end. And sometimes it's not going to be gene editing. It can be many different things. It can be agronomic processes. In fact, many analyses of yield improvements over the last 50 to 100 years suggest that about half of the yield improvements are related to improvements in agronomic practices, and half with plant genetics. And in fact, often the two go hand in glove together. So you get changes in the plant genetics which enable changes in the agronomic practices, or changes in agronomic practices force changes in genetics. And one of the classic examples of this is analyses of the extraordinary increases over the past 60 years or so of corn yields in the Midwest of the United States, where the percentage increase annually is about double that for almost any other crop in any other system. Spectacular impacts. And a lot of that increase in yield can be attributed to an increase in the planting density. And the increase in planting density can be attributed to a change in leaf angle. So the leaves now grow more vertically, so the plants can be planted more densely. The light can be harvested more efficiently as it penetrates down through the canopy, and you can get, yes, a doubling in yield from a doubling in planting density. But it's having an effect because of a change in leaf angle. I must admit, critically assessed that study, but I think it is a very solid piece of work. So agronomy, breeding. I've actually put in another thing, which is some of my own work at the moment, where we've been doing some really interesting work I'm very excited about in my own research laboratory with grafting. So of course, grafting isn't relevant for example, the cereals, but it is relevant for a lot of crops, and not just perennial crops, remarkably. So in some areas, like in some parts of Western Europe, as much as 70% of the tomatoes being grown are grafted. And this is leading to very big increases in yield and extraordinary acceleration in breeding programs, because you can have a breeding program to improve the roots, and that's quite distinct from the breeding program to improve the shoots and the fruits on those shoots. And I think grafting is an area where for a wide range of horticultural crops, very big increases and very big impacts can be quite rapidly delivered. You can also be more bold, if you like, with the development of the rootstocks. You can get, relatives that are quite distant, and so it helps you access, for the development of rootstocks, it helps you access a much wider germplasm than might be otherwise possible. Whereas if you're constrained with, the need to have these fruits which are going to be acceptable for the market. And then, of course, there's the more high biotech approaches, such as development of transgenic crops, and of course the development, which is the primary topic of this workshop, of gene editing. And gene editing, of course, is extremely powerful and allows you to improve your science faster, but also can give you, I think, potentially, at any rate, much more rapid delivery of crop improvements. I have to admit, I don't know what the public acceptance and the legislative, legal acceptance of gene editing is in many countries. The European disaster is not helpful, but I think we should still forge on, nevertheless, in using and hopefully deploying gene editing, because it also, in any case, it allows for good science to be done faster and more efficiently, and thus genes and alleles of genes to be more rapidly identified. And then the last step in this process of crop improvement is delivery. So don't just do it, don't just write academic papers, but also absolutely get what you have done actually into the farmers' fields. And this is something that's extremely important. It's complex, it can be difficult, it can be risky, but we've got to do it. Okay, so I'm going to just go through these 4 points again, and then just fold in materials from my own research. So it becomes a little bit more Mark Tester focused, but I'm just doing this just by way of example, and it's stuff that I know a fair bit about. So identifying the target, for me, I'm very interested in salinity tolerance, and the context of this is that agriculture globally and regionally is by far and away the largest single user of water by humans. So about 2/3 of all the water that us humans use is used for agriculture, and that water used is producing about 1/3 of our food. Some estimates make it even more, up towards 40% of all our food is produced under irrigation. So a lot of our food is produced under irrigation, and this is using a massive amount of water that humans use. And the estimates are a little bit wobbly or loose, shall we say, but in the order of half of all the water that is used for irrigation comes from groundwater. And the very alarming fact, the empirical observation, is that every major aquifer that is being used for that irrigation is being depleted. In other words, we are unsustainably mining groundwater resources for the production of a large fraction of our food. This is a classic definition of an unsustainable practice. It has to change. It would change when the water runs out, so it's going to change. And what we really have to do is examine extremely carefully how we are using our groundwater resources. We use them, we should use them. I'm not going to go around saying we should stop pumping groundwater, but what I am saying is that what groundwater we are pumping, we should be using very, very wisely and try to maximize our benefits from the groundwater that we are using. And of course, if every groundwater source is being depleted, we have to reduce the rates of pumping of that groundwater if we want to be continuing to use groundwater for a longer, prolonged period of time. And one way, there are many ways to do this, but one way which just draws on some of my technical skills and experience is to see if we can substitute some of the fresh water that we're using in agricultural systems with salt water. For every drop of fresh water underground, there's another drop of brackish water, plus of course there's massive amounts of water in the sea. And it would be great if we were able to use technologies to unlock seawater and other water we currently cannot use to try to contribute to global food security. So that's the target that I've identified, and I think it's in particular, living in Saudi Arabia, it's a perfectly reasonable target for my work. So then the second thing is, how do we address the target? We have to identify the systems, crops, processes, genes, alleles of genes for seeing if we can increase our use of saltwater resources. And for me, if we're going to unlock something like salt water, we need a key. And this key, I think, is going to be very often technologically based. And we have now a pair of beautiful technologies, very powerful and exciting technologies, to try to help us unlock seawater, and I think for a lot of you in the audience, help you address your targets that we're all wanting to address. And those keys for me are what I call the new genetics. Genomics is amazingly powerful and is turbocharging genetics. And so just as medicine is facing a revolution powered by genomics, so too is agriculture. And hand in glove with the genomics charged genetics, we can also now turbocharge our characterization, our phenotyping of the plants using a combination of machine learning, computer vision, these Internet of Things, these IT in particular, IT-based technologies that are driving phenotyping as well as genomics, we should acknowledge. These really do provide us with serious opportunities for major new innovation. And in particular, I think it gives us huge opportunities to unlock wild germplasm and access whole new ranges of genetic resources, which can then be delivered using a whole range of technologies, including genomics, accelerated breeding, marker-assisted selection, and so on, all the way through to gene editing, the topic of this workshop. Okay, so yes, I just have a picture here of a drone carrying cameras over, this is a field of quinoa that we were phenotyping at one stage. So these are very useful, I think, technologies which I think we really can deploy a lot more. So when we're unlocking wild germplasm, for example, or I would argue in particular, we can do this in various ways. We can, for example, get the crops that we have already existing and make them more salt tolerant for my little set of work, and drought tolerant or heat tolerant or whatever else it is, disease resistance that we're wanting to target and needing to target for our particular problem. And that's one good way to approach things. Get the crops and then improve the crops that we already have. And we can bring in genes, resources from wild relatives. And there is an alternative that we can make, which I'm increasingly favoring as I get older, and that's to be more radical. It's interesting, young people are more radical, but I've got more radical as I've got older. And what I'm thinking that really is possible and very exciting is to get wild plants that are not crops, but already have extraordinary abilities to grow in my particular work, saline environments and even seawater, and turn them into crops. So in other words, implement what I would call neo-domestication program. And here I've got some examples. This is quinoa, which we've been doing some work on. Fabulously salt tolerant plant. It's already a good crop, to be fair, but it does need more domestication. So I'd say it's only partially domesticated and has many traits which can be improved. And here's another plant, which is a wild tomato growing on the rocks in the Galapagos Islands, being splashed by seawater, and for which salinity tolerance traits can be integrated into commercial tomatoes. Here's which is one way of doing it. So here we've got just example of some work that was actually published many years ago now, where Rana Munns came to my lab when I was actually still in Cambridge in England, and she had some bags of durum wheat seeds in her hand, which had much lower accumulation of sodium in the shoot. And we identified the gene that was responsible for that reduced shoot sodium accumulation, and in particular Rana moved the gene, integrated the gene into commercial durum wheat, and this led to some very interesting field trials where the integration of the single gene had no yield penalty in low sodium parts of the field, but led to a very substantial increase in yield in the very saline parts of the field. So it's great work, and it was published in Nature Biotech. It was very, very nice. Why am I not more happy about that? I'm very happy with the science. It was very good. It turned out actually that the gene actually came in from a breeder crossing in diploid wheat into tetraploid durum wheat from wild wheat, from the A genome donor, into durum wheat back in the 1960s, and they were doing that to bring in new alleles for rust resistance, and in the process they accidentally brought in a gene that was helping reduce the root to shoot transfer of sodium, and that gene, for whatever reason, was absent in the original tetraploid durum wheats. So it was in fact a wild relative bringing in a new gene, adding a gene into the genome, which led to this salinity tolerance. So I'm not dancing around with great joy from these results, because the impacts of the gene were only present, it turned out, and to be fair to us, I don't think we could have predicted this, but it turns out that the benefits from this gene are only present when the plants were growing in very, very saline parts of the field. And so that 25% yield increase was happening when the yield had already been decreased by 2/3. So in these control parts of the field, the yield is about 3 tons per hectare. And in the slightly saline parts of the field, it was more like 2 tons per hectare. The gene was having no impact, negative impact, which is great, but it didn't have any positive impact either. The only time it had a positive impact was when the yield was reduced to about 1 ton per hectare. The plants were half dead. And then the gene was having a benefit, which is fine, but that's not going to help feed the world, really. It's not that big an impact, because really this is not part of the system which is intensively cropped or should be intensively cropped at all. So we start to look at other traits, and I moved to Saudi Arabia. It was almost 9 years ago now, and we started to open up a whole series of projects where we're looking at a whole series of other traits looking at the impact of sodium on not just sodium accumulation like in that previous work, but in other traits which could also be important in contributing to salinity tolerance. This is one piece of work that I'm very proud of, done by one of my many Saudi female PhD students. This is Nadia Al-Tamimi, who actually now works in my company. She was looking at the effects of salinity on transpiration use efficiency, and did this in a mapping population of rice. She was looking not just at transpiration use efficiency and the genetics of that, which is hopelessly complex. She was doing something important. She was looking at the effect of salinity on transpiration use efficiency. So she wasn't just looking at the genetics of transpiration use efficiency, she was looking at the genetics of the effect of sodium on transpiration use efficiency. And bang, came out with a very clear locus, a very strong impact from a single gene on the effect of plants to maintain their transpiration use efficiency in saline conditions relative to non-saline conditions. So it sounds subtle and complex, but I think it's quite important. And we're now, of course, trying to identify the gene responsible underlying this locus. And it's very interesting in that locus, which has got about 20 candidate genes in it. There's 5 tandemly repeated, annotated as protein kinases of unknown function. So we're starting to do the hard work, frankly, to characterize the genes in this locus. Okay, so those are just 2 examples of how we've been trying to get commercial crops and increase their salinity tolerance by looking at traits that are hypothesized to contribute to salinity tolerance. Now just want to just quickly introduce to you another very interesting area, an area where gene editing can have some important contributions, I think, which is for the domestication of plants which are already very salt tolerant, such as in this picture where we've got quinoa, which is a classic crop for marginal lands, where people, in these very marginal environments on the altiplano of Bolivia, they say we have nothing else but quinoa. It's the only crop they can grow. So they think it's the only crop that they can grow, and in very, very sparse field conditions. It's also a crop which has massive yield potential, so you can get remarkable high yields, such as here in the Tibetan Plateau in southwestern China. It's also a crop which is notably very highly nutritious and is the only grain that provides a balanced nutrition, a balanced portfolio of amino acids as well as minerals and vitamins. So it really could be a crop for the future, if only we could improve its traits, and a range of traits that need to be improved are really some pretty fundamental agronomic traits. So to turn it into a plant that's sort of more like wheat, I guess, is the paradigm I have to admit I come from. Not necessarily the correct one, I should say, but it's certainly where how my head works. You need to have larger grains, higher yield, better quality. But I think yield stability is also an extremely important trait. The plant needs to be grown and more securely, if you like. And we need to improve early vigor, need to improve dwarfing, reduce branching. And because it's a dicotyledonous crop, it has a much lower portfolio of herbicides that can be used. So making the plant herbicide resistant is another extremely important target. Again, that could be the best way at the moment to do that is through mutation breeding, but it's also possible, of course, through gene editing. The main crop that we're looking at, the main target trait, that we're focusing on at the moment is heat tolerance. Now the plant is famously quite sensitive to heat, even though it's quite tolerant to drought and salt, and it's a pretty good plant in many ways, but it is sensitive to heat. And so we're trying to introgress heat tolerance from a wild relative, which is extremely heat tolerant. And we've got Chenopodium hircinum, which is the immediate tetraploid precursor to quinoa. And there's some very heat tolerant examples of this plant growing in northwestern Argentina. We've collected the seeds, sequence the genome, do all that sort of stuff, and start to characterize the heat tolerance. Okay, and of course, if we're going to improve quinoa, we need to have a good quality genome, and you need this for your gene editing, for a lot of things. I find this as part of the foundation upon which a crop improvement program can be built. So we've now done a version 2 of the genome, which is largely released. We haven't got around to publishing it, but we have released it, and a lot of people are already using the version 2 genome. It's much tidier than the original, the genome originally published. We also have the 2 subgenomes identified, 2 diploid wild relatives, and again there's some very nice syntenic alignments, and we're able to identify really just a very small number, maybe 3 or 4 major chromosomal rearrangements, and otherwise the plant is, we've got a very well behaved, if you like, genome. It's an allotetraploid, an ancient allotetraploid. And then we can start to do some population genetics as well. So we've resequenced about 1,000 accessions and can group these into, the number's a bit wobbly, 6 to 8 different major subgroups. And then within each of these subgroups, we can identify lines, individual lines, which have got very high purity, genetic purity, and also a reasonable phenotype, because we've been phenotyping them in many locations for many years now. And we've now got, we're now de novo resequencing 8 accessions for, I guess, doing a proper metagenomic study. So this is like the next level, which becomes increasingly important. So here are here's a phylogenetic tree identifying the relationships among these accessions. The quinoa is divided into 2 major groups, the lowland and the highland groups, and then within each of those 2 major groups there are, who knows, some set of subgroups. But this type of analysis can be really valuable for breeding programs. If you're searching for more genetic diversity, or diversity in some sort of trait of interest, then you can really reduce the number of plants that you have to screen by taking a sort of, if you like, an intelligent subset from right across the genetic diversity spectrum. It's also quite interesting to note, just on a more academic comment, that the wild relatives in this phylogenetic analysis come out between the 2 subgroups. And at the moment, we're certainly thinking that quinoa has actually been domesticated independently twice, once in the lowlands of Chile, and then a second time in the highlands, possibly even in northwestern Argentina or southern Bolivia. We don't know. This is speculation, but it's certainly a very interesting observation, and it's also useful for breeding considerations, crop improvement considerations. Again, we're looking at breeders want more diversity in injected, or particular traits injected. So if you've got something which is like a real pure highland line, then maybe it's worth investigating for genetic diversity from the lowland lines. Okay, so this is just re-saying what I've just said. My apologies, I got ahead of myself. Linkage disequilibrium when you're a breeder is also very important. I'm not a breeder, by the way, I'm a plant physiologist, if I'm anything. But it's quite interesting to see that there's a very big difference in the leakage disequilibrium between the lowland and highland groups. And of course, the molecular basis for that is unknown, but it would be very interesting to know. Okay, so we do genomics, we have opportunities to do phenotyping. So now we have opportunities to do quantitative genetics in the field, and using drones to not just make pretty movies, but also to quantify the growth of the plants. And you can do non-destructive monitoring of growth and the response of the plants to their environmental conditions in a way which is much, much cheaper and much more powerful than has ever been previously possible. So we can now do fields with large numbers of accessions and then phenotype them using drones to characterize traits. And we're now starting to do this in a range of different locations, and that's good, but it's also dangerous, because if you've got lots of different people phenotyping, you can get yourself into quite a lot of trouble with language, for example. And I don't just mean Urdu versus Spanish or something. I'm also talking about how we are defining the phenotyping. Now, I'm emphasizing the value and future power, even current power, of drone-based imaging, but we also need to phenotype using traditional methods on the ground, and these methods need to be internationally standardized for crops. We've got a new crop here, quinoa, so we've actually just published a global consensus. This has got over 50 authors, I recall, on this paper, where we tried to get a global consensus for the phenotyping, and have with that cards that you can print out, laminate, take into the field with you. Here's the paper. So there was all of these primary authors, and then there was also about another 50 people in what we call this phenotyping consortium, so people who basically signed up to this method of, or these standards for phenotyping. I keep mentioning these drones, but they really are very, very, I think, powerful, and I think they're going to become, well, they are becoming much, much cheaper and much, much more powerful. And with software, you're able to start to do, with the development of software, we're starting to be able to do, frankly, remarkable things. So this is not a picture of a field. This is a 3D artificial reconstruction of a field just built in the computer from the drone images. The drone's flying over. You're not doing any of this 2 cameras fancy stuff or anything. This is just from single drones. Because the drones wobble, they pitch and yaw as they're flying along. Just from those little, if you like, errors, you turn those errors into an advantage, and from that you're able to reconstruct in 3 dimensions the field in color, or actually hyperspectral, so you can go across a very wide set of bandwidths. Any rate, I must move on very quickly. These sorts of things are powerful and interesting, and I think are a future to help us identify variables which can then be used to predict yield maintenance, and all sorts of other traits. I think that's quite useful. I'm going to move on through this. We've also been looking at photosynthetic traits. Another one of my Saudi female PhD students who's finished and now doing a postdoc in South Africa. You have lots of things you can measure now much, much more rapidly than previously. So you're getting lots of data, and that can turn into a nightmare for students. And so data handling is becoming quite important. And my students all got together with one of my postdocs, Magda, and they developed an app called MV App. I really commend anyone in the audience who's doing science actually to use this app. A lot of people are now. Magda led a whole bunch of students to develop this app as a team, and I think it's a really good thing. So do have a look at that if you're wanting to do lots of phenotyping. So you phenotype, you genotype, you can then start to do association genetics. I'm going to spin through this now because my time is up. I think I've said enough and given you the ideas at any rate. You're able to identify loci, genes, the alleles of genes, which can then be your targets for crop improvement. if you've done those processes before. Remember, you have to have the context to make sure that what you're measuring is relevant to ultimately crop improvement for your country or your region. We're trying to near domesticate crops, plants, extreme plants, and we're working with Salicornia now. It's a very, very interesting plant. There's lots of genetic diversity. It has genomes that are moderately tractable, and we've been starting to do some, in this case, a proteomic study to identify the proteins responsible for the extraordinary ability of the plant to have extraordinarily high concentrations of sodium inside its vacuoles, which you can go all the way from a Salicornia genome to a yeast plant. That was all done by one PhD student. Okay, so this next bit's the big bit, and that's your course. All right, I'm going to leave you with this. I'm not going to talk about the appropriate technologies. This was meant to be an introductory talk to give you some context for then you to go off and not just in inverted commas do gene editing, but gene editing that can really count and really make an impact. That's what I would love you to do. I'm just going to finish by a quick word about targets and delivery. We're really now, in my career now, we're really focusing a lot more on delivery, and we're focusing a lot more on covered agriculture. And covered agriculture, I think, is extremely important, because one of the biggest issues now globally, there are two big issues globally in agriculture, in my analysis. The first is the sustainability of the agricultural systems. And the second is the nutritional value of the food. So at the moment, we are already producing more than enough globally. I wouldn't say within a particular country, but globally, we are producing way too much meat and starch already. But we're not doing it sustainably, and we're not providing sufficient nutrition, vitamins and minerals for people. And for me, those are two massive targets, which I would urge all of you to be seriously considering. And by this analysis, which was done by the Eat Lancet Commission, a very, very authoritative report published a couple of years ago, they're saying that for targeting 2050, and if we reduce waste we haven't even talked about at all, we actually are already growing enough cereals globally. We're already growing enough cereals. By 2050, that's a long time away, we don't need to increase cereals production. What we do need to increase in particular is the production of fresh fruits and vegetables. And globally, I'm not talking about locally, but globally we actually need to reduce red meat production substantially. Okay, over halving red meat production globally for a healthy diet, if we also not eliminate waste, but halve the waste. So it's a very, very, very interesting analysis. At any rate, increasing fresh fruit and vegetable production really means that we really need to increase production of these locally, because these are things which have a very high carbon footprint and very high cost for transport, because they're not easily stored. And so for me, developing greenhouses in desert environments in which fresh fruits and vegetables can be grown economically viably is really important, and with a reduced environmental footprint. So we're trying to deliver some of our research by growing salt-tolerant grafted tomatoes in a greenhouse using diluted seawater, in a greenhouse cooled with seawater, where the roof is cleaned with no water. We end up developing a system where, there's between, 1/10, or let's say roughly 1/10 of the water required for the production of the food in this system compared to a normal greenhouse or open field system. So we're bringing together engineering and plant science with IT-based systems, which are really powerful, to develop these saltwater greenhouses where the freshwater footprint is much massively lower than the freshwater footprint in a normal greenhouse or in an open field, and that comes with no increase in energy consumption, if anything, a decrease in energy consumption. So we're actually now developing a whole stack of technologies, engineering, IT, and plant science technologies to try to deliver some of our research in an economically viable way that will self-propagate. It will be stolen, it will be copied, and that's great. Okay, so I'd just like to finish here by, of course, thanking all of the people who I've been collaborating with over the last many years. I'd like to thank my lab. This is how we used to meet. This is a photograph from 2 years ago. This is how we meet now, very sadly. But I'd just like to thank you all very much for listening. Thank you. Hello? Hello, is that okay? I can't hear anybody.
Mark, thank you very much for your presentation. It was very comprehensive and again, a very expensive one as well. Sir, with your permission, I would like to take some questions in the audience as well as from the virtual participants as well. So do we have questions from the in-house? Yes. So we have one question. Let me just, madam, can you please come here for the question, please? Yes, please.
Hi, Mark. I'm Shadi Hatta from and I'm responsible from wheat transformation and genome editing. Of course, my question is about in wheat. And have you carried on your wheat research, or you just stop after those interesting results? I think the results you present are very interesting. And have you considered to do any wheat transformation?
I'm sorry, I didn't quite catch your question. My apologies.
Mm-hmm. Okay, because your first results were in wheat research, but after that you start represent other results as well in salt tolerance. Is there any extra results you get from wheat, or you stop the work in wheat, or have you got any target for the wheat?
Yes,
or
yes.
the salt tolerance?
Sorry, that's an excellent question. Very good question. No, I'm afraid, well, coming and moving to Saudi Arabia, and then realizing that really wheat is not a good crop to grow when you have no sustainable water sources. I stopped working on wheat. Yeah, it's a pity because I was doing quite a lot of work on wheat, but I left my laboratory behind in Australia and it's still working, it's still operating. I'm very proud to say, under the leadership of Stuart Roy, who was my deputy. So I liked my more egotistical moments. I like to think, my legacy is living on, but in Australia through Stuart Roy. But of course, he's developed into a mature, independent researcher, of course. So Stuart, I would commend you look at the work of Stuart Roy, who I know is doing work that is definitely focused on true delivery as well, not just academic work. So yeah, look at the work of Roy et al. And you'll see there were many papers with my name and him publishing together. I think that's faded away. I'm not sure if I am still on some of his papers.
No, it's great. Thank you. Thanks for the answer. Just I'm interested because if you got interesting targets, you can now transform different wheat varieties because you're interested in the room wheat. So that's why I asked.
Get in touch with Stu. Get in touch with Stuart Roy, and he'll be your best person for wheat specific targets, and he'd be interested to talk because I know he's still using biotech approaches. But yeah, I've moved more on to quinoa and tomatoes and salicornia. They're my three main targets now.
Okay, thank you, Mark. Thanks for the answer. And say hello, Brenda, from us.
Ah, yes.
Yeah, we are connected together from Johnny Center.
Fantastic. Thank you.
Yes, sir. Thank you very much, madam. With this, I think we have question from Dr. Shahid Mansoor as well, sir.
So, hi, Mark. It's always a pleasure to listen to you. Very philosophical and very close to what we want to achieve. So I think this is a very good lesson that you have given to us. My question is about the covered agriculture. So obviously, you said that this is the future of agriculture, food security. So what about the energy costs? I mean, that's one of the limiting factor probably when we are going for covered agriculture.
Actually, the energy costs are not too bad. The main energy costs are for pumping water and for turning fans. You certainly do not want to use mechanical cooling, like a refrigerator-based cooling. Then your energy costs go up through the roof. And you certainly do not want to do this current fashion of vertical farming. This is insane because the energy costs are huge because you're using artificial lighting. So in the covered agricultural systems, you don't use artificial lighting, you don't use, mechanical cooling, but you just, in inverted commas, need to pump water and turn fans. And sure, that uses energy, but it's not too bad, actually, Shahid. One thing which we do, which we've actually innovated with, is we're using fans powered directly with DC current. Okay, and this is important because, and it took a small modification of the system, and that's all that's been required. We did this all in-house, and with the DC fans, we can use solar, and we can directly connect a relatively small solar panel to a fan, and that provides enough electricity to turn that single fan. So you have a direct fan with its panel, and you don't need an alternator, so it's cheap because you can use DC to drive the fan, and you don't need a battery because when do you need the fans turning? You need the fans turning when there's a big heat load in the greenhouse. When is there a big heat load on the greenhouse? When the sun's out. So the solar panel, it's so beautiful. So the sun is out at exactly the time when you need the fans turning and the pumps pumping, because you need the pumps to pump the water over the pads. This is all evaporative cooling. And so we can actually economically have, maybe 50-60% of the modest energy requirements of the greenhouse being covered by the by solar panels and a fairly small area of solar panels. So you end up with the primary limiting factor, I think, for a lot of systems actually being the capital cost, the upfront capital cost. That's the major problem for greenhouses, which I know is a non-trivial problem. Okay.
That's another cost. Okay, thank you.
With this, do we have any other questions? I think we should also take one question from.
Hi. Okay, my question is, how is the acceptance of people in the UK and Europe regarding genetically modified crops? There are people who are against these genetically modified crops, and we are doing a lot of research, but sometimes we cannot apply in the real life. So how is the acceptance of the people?
Yep.
Okay, thank you.
Yep. I think you know the answer to this question. The public acceptance is still very low. There's a legislative constraints which are crippling in Europe, and I must admit I'm very depressed by this ongoing unscientifically based resistance to the uptake of genetically modified and gene edited crops. What I find quite interesting is that people seem to have, the broader public have seemed to have turned to science very quickly for the pandemic and seem to be putting a big trust in scientific evidence for the management of the pandemic. And so maybe that will increase people's ability to accept scientific evidence for other areas in life, such as genetically modified crops. This may be a vain hope, but I think the pandemic has done a lot of good for public acceptance of science and people realizing that science is actually a very, very valuable thing. So I hope that acceptance of gene-edited crops will increase because of people's higher realization of the value of science. But at the moment, unfortunately, it's a good question, and unfortunately, the public acceptance is still low. That's why I'm doing a lot of work with marker-assisted breeding and genomics-powered breeding. But keep doing the gene editing, all right? It's more powerful than anything, and surely there will be public acceptance increasingly over time.
Thank you very much, Mark. And if you allow me, I will take one last question from our virtual participant as well. So we have a question: what if we are making starch and meat in bulk? The people who are, sorry, let me read another question.
What if we are making starch and meat in bulk? Well, I don't understand the question. So
Mark, let me take another question. I think that was it wasn't very clear to me as well.
No. I mean, locally, we do still obviously need meat and starch production, but like a country like Pakistan has done brilliantly and produces enough starch for its population. It exports starch, which is just amazing. It's fantastic. But my concern is that it's not environmentally sustainable, and it's also, what's much more important than starch now in Pakistan is the micronutrient deficiencies. Now, they can be partially addressed through meat production, but they can also be partially addressed by improving the production of quality fruit and vegetables, or fruit and vegetables that contain these micronutrients that are so needed.
Thank you very much, Mark. And with this, again, I would like to thank you very much for your time, and also thank you very much for helping us in submission of this project and getting this project submitted. And again, thank you very much for your time today.
Absolute pleasure. Thank you, and very best of luck for your workshop.
Thank you very much. With this, we move on to our next speaker, Dr. Hensel from Germany. The title of his talk is Gene Editing in Crop Plants, Development Towards Commercialization. I would request Professor Dr. Shahid Mansoor to introduce our next guest speaker.
Thank you, Hazima. It's a pleasure to introduce Bruce Hensel. He has visited us in Pakistan when we were holding the first meeting. Bruce Hensel has main focus on targeting genome editing modification of plants. This includes classical transgenics approaches, but also the use of new method of targeted mutagenesis, better known as CRISPR-Cas technology. The aim will be to establish and develop suitable vectors and tissue culture system for plants, especially that are in the research focus within CIPLAS, which will enable genetic transformation and thus genome modification. With this brief introduction, I welcome Bruce Hensel for this talk. Thank you. So, is he online?
Yes, he is on.
So, yeah, I mean, you can start talking, Bruce.
Yes, thanks for this nice introductory words. And I also apologize for not being in person in your conference, but you may understand that at these times traveling is not that easy, and it's also during the semester. So that's why I decided to participate online. And I want to thank Mark for nicely setting up the stage for my talk. And I will share my screen now. So I hope everybody can see it in presentation mode. So if I do not hear somebody complaining, I will continue. So my talk is about gene editing in crop plants development towards commercialization. Sorry? I cannot.
Dr. Hensel?
Yes.
Dr. Hensel, we can clearly see your slides and hear you. Please continue.
Okay, thank you. Yeah, so as mentioned, and also introduced by Shahid. So I recently moved to the Heinrich-Heine University in Düsseldorf, and I'm associated member of the Cluster of Excellence of Plant Science, which is also has its main office in Düsseldorf. So before, I have been working many years in the IPK in Gartersleben, and now I establish the Center for Plant Genome Engineering, which is now located to this new complex of buildings at the Heinrich-Heine University campus. And the topics of my center are, on one hand, transgene technology development. So I'm interested, or we are interested in chloroplast development. I have a long-lasting interest in molecular farming. So this means producing recombinant protein in plants. And as I'm now associated also to the Institute of Plant Biochemistry, headed by Andreas Weber, who is mainly interested in photosynthesis. So now I also focus on minimizing photorespiration to make crop plants more efficient. So what plants we are working on? So my center is mainly establishing and using methods for plants which are in the focus of the Cluster of Excellence in Plant Science, C+. So I have a long-lasting history in working in barley and corn, and recently started also to work on sunflower. Then we are just starting a project on amaranth, which nicely fits also to what Mark has presented before, that we can use our plant species in order to use gene editing or genome editing to improve traits to make them nice crops. As I'm working now on the switch of C3 to C4 photosynthesis, so I'm working on Moricandia, which has family members, which are C3, C4 intermediates, and so therefore we would like to identify genes which are responsible for this switch in the same direction. So I'm working on a calm plant, which is Talinum, and we use model plants like tobacco and Arabidopsis. So just to give you a brief overview, what I'm doing in my center, and now coming to my talk. So you might be aware that for genome editing or gene editing, we have mainly 3 platforms, which are illustrated here. So we are starting, or the technology starts with the use of zinc finger nucleases. Then afterwards, transcription activator like effector nucleases become useful. And the latest 2 here in this direction are the CRISPR-Cas technology. All these three technologies has in common that they are a two-component system, that they have one part which is responsible for targeting, while they have a second part which is responsible for induction of a double-strand break, which then cause modifications we are interested in. While the upper two are protein-based, so here you have peptides, either that three peptide or one zinc finger target a triplet of DNA, while for the talents you have peptides which are specific for individual nucleotides. The CRISPR system is based on RNA. So in principle, you have two components, one for the targeting and one for the induction of the double-strand break. Coming to the CRISPR-Cas technology specifically, so here biotechnologically, we're using components of a bacterial immune system. It has its beauty as it seems to be very universal so far in agriculture and medicine, and every application and approach has been used. It was successful, and so maybe this was one of the reasons why it became acknowledged by the Nobel Prize in 2020, because it really revolutionized the way we are able in targeting genes in particular. And as I'm working since a long time in this topic, so it's really amazing how precise we can do gene modifications these days. So as illustrated here, and as mentioned before, we have a two-component system, and I can't go into much detail as this will be also part of the talks which are coming afterwards. So I just want to briefly mention that this two-component system induce a double-strand break, and as these are also naturally occurring, so cells have developed their own repair mechanisms in order to cope with those double-strand breaks. And there are two main pathways which cells have developed. The most common one and easy one is the non-homologous end joining. So where you just stick together the two loose ends in order to repair. And this in the majority of cases is error-free, but in some of the cases you can have deletions or additions of one or a few nucleotides. A bit more complicated and less frequently used are homology-directed repair pathways, which usually use donor DNA like the sister chromatid or some synthetic DNA, which you can provide in order to use homology-directed repair mechanisms. And these are, of course, needs more energy, and so therefore this is not the preferred pathway. But these are the two main pathways which are used after this double-strand break happen. So as mentioned before, as it turns out that this gene editing technology is very universal. So over the years, a lot of different studies have been accumulated. And here in this table from a recent paper summarizes which categories and which plant species have been using gene editing. And so you can see the list is already quite large, covering feed crops, fiber crops, food crops, crops for industrial use, oil crops, and ornamental crops. So it's really a huge list and shows the power of those technology. And if you are looking for the traits which the people are targeting, so it's funny enough, at least for me, so that they are still the same as the Andrian farmers 10,000 years ago started to look for, because these are also summarized here on this picture. We are still looking for a bigger or higher number of fruits. We look for the fruit color, we look for grain size and so on, for reduced dormancy. So the traits we are working on are still the same as 10,000 years ago. But now we have tools in our hands which speed up the process and can be done more precisely and specifically. So in particular, if you then look how many studies have been published per plant species, so you can see, of course, that the two main model plant species, one Arabidopsis and the other one already a crop plant, rice, really dominating the field and showing also the power of the system. Yeah, so in rice you have an incredible number of 500 publications. So this was just by the year 2019. So for sure we are almost double this by the year 2021. And there are others which are not covered that much yet. And if you look again into what topics the people are focusing with those studies, then you see that the majority, and as also outlined by Mark, are the agronomic value. There is food and feed quality, taste, color, and so on. There is, of course, the impact of biotic stress tolerance with the spread of diseases to places which they have not occurred before due to the climate change. And there are other topics like herbicide tolerance and so on. So if you think in terms what developments we need to go with the gene editing plants for commercialization. So I listed here some points which in my eyes are needed. So on one hand, of course, you need your genome editing reagents. Then, of course, you need transformation and regeneration protocols in order to generate your gene-edited plants. Then, of course, you need sophisticated detection methods in order to identify those events which have been altered. And last but not least, of course, you need freedom to operate and legislation, which was also a point in Mark's talk and also already in the discussion. Yeah, so of course, we need also the legislation which support the development of those plants. And starting with the first point, so there are several ways how you can assemble your gene editing units. So most common are either plasmids or linearized fragments where you assemble your promoters, your endonucleases, guide, and so on and so forth. And you will become more introduced and get deeper insights into this for sure in the coming lectures. So then people can also use RNA. And last but not least, here also you can use pre-assembled ribonucleoprotein complexes, which means that you already in vitro assemble your RNA and your endonuclease. So then, of course, after having those gene editing units, you of course have to think about how you can transfer those into your plant cells. And where over the last decades developed a lot of tools which can be used in order to introduce your gene editing units. So most common for plasmids are Rhizobium radiobacter, which is maybe more common as Agrobacterium tumefaciens to you. Then there are, of course, biolistic methods where you coat either gold particles or others with your DNA, RNA, or your ribonucleoprotein complexes. You can use electroporation, polyethylene glycol. Recently, also nanoparticles become very common in the delivery of those reagents as listed here. Yeah, so this list is not complete and can be extended, but this mainly lists all those delivery tools which scientists have in hands these days. Then coming to the next point, of course, after having your units and an idea how to deliver these into plant cells, then of course you need to make new plants out of those transformed or altered cells. And then there are main differences between monocots and dicots. And here I listed at least some explants which can be used for the generation of those gene-edited plants. So in the dicot, it's quite easy. So you can use either leaf disc like for tobacco or floral dip, as is common in Arabidopsis thaliana. You can use hypocotyls or even protoplasts. These are explants which had been shown useful for the generation of new plants. In monocots, which I have been working on over the last 2 decades, it's more complicated. So here we have those explants which you can use. The main working horse are immature embryos, where routinely protocols exist in barley, in wild barley, Rhodium spontaneum, in wheat, in rye, in triticale, in maize, and in teff. My former group leader, Jochen Kumlin, has developed a very unique system which is based on embryogenic pollen cultures. This has the advantage that you are dealing with a haploid genome and your modifications become immediately obvious because you can make already homozygous material. In terms of genotype dependency, which we have in most of our transformation protocols, isolated ovules have been shown by us and others that they give less genotype dependency. And here protocols are published for barley and wheat. And you can use also some shoot segments. And this we have shown for Brachypodium. So as mentioned, I was mainly involved in the development of protocols for barley, where we used immature embryos, and over callus induction and regeneration we come to plants which then can be confirmed to be transgenic. And on average, we had an efficiency of getting 20 primary transgenic plants per 100 immature embryos inoculated with agrobacterium, so which is quite good efficiency. And over the last 20 years, I have been involved in nearly 1,000 transformation experiments, generating more than 20,000 primary transgenic plants. So as we were also interested in other cereals, so we have also published protocols for wheat and triticale and corn, and also working on other in this direction. So then next, after having your regenerants, of course, you are interested in the detection of your gene modification events. And for this, the most popular is sequencing of PCR products. Either you send them directly to sequencing, or after a short ligation and amplification in bacteria, you can isolate your plasmid DNA and send for Sanger sequencing. And a chromatogram of such an gene-edited plant can look like this. So here you see an example for a diploid plant like barley. So on top, you see the target sequence of our CRISPR approach in red, and the protospacer associated or adjacent motif PAM is here highlighted in blue. And here I indicated the place where we expect to induce a double strand break. And then until here, you would have a clean sequence, while after that, if you have a modification, then you get such a weird chromatogram. Maybe some of you have seen already weird chromatograms, but it should be clean until the place where the double strand break occur. So it can look like this. And so as this can look even more weird, as our primary regenerants are usually chimeric. So this means that each cell may have a different mutation. And in a diploid organism, this could be, in theory, 2 different mutations within even one cell. So that's why this can become a mess. And there are online tools which solve those problems. One of them is called Tide, and you can use this online tool in comparing your wild type sequence with your mixed pools. And then the program make a decomposition of those chromatograms and then give you percentages of the reads which are in your sequence. So in this way, you can get an idea what mutations are in your plant. So usually you test leaves of your primary regenerants, which does not necessarily mean that it also go to the germline, but it already shows that something is ongoing in your plant. So then another possibility, of course, is deep sequencing. So as people also use protoplasts in order to test their gene editing reagents, so you can then use next generation technology where you usually start with making a library of fragments of your DNA and then label them accordingly and sequence. And then, of course, you need strong bioinformatics to make sense out of the many reads and to give an annotation and assembly of those fragments to give sense out of it. And then at the end, you get also a table which contain then the frequency of your insertions, deletions, and you get an Excel table where you really can see which modification occur in what frequency. So this is also as the prices for next generation sequencing are dropping. So a very nice tool as well in order to get a deeper information into what's going on in your experiment. So then, of course, you need freedom to operate. So what does it mean in my place? So I would start with one thing, so that as also was already a point in the discussion, that people are, at least in some parts of the world, having problems with having transgenic plants. And one thing of overcoming this is to use RNPs, ribonucleoprotein complexes, as those are just RNA and protein, so they do not transfer DNA into a cell. So this would be maybe an elegant way in order to circumvent this. Also in Europe, I have to add, this is still considered to be a GMO. So what's going on? So you can order your Cas protein and your guide RNA, you can assemble in vitro and then transfer those into your cells. And here I will give one example of my former postdoc, Martin Becker. So he transformed immature embryos of barley with an RNP complex targeting a barley gene, which is involved in beer quality. So here you see the target site. In order to be sure that we have a successful transformation, he co-bombarded with a red fluorescent protein, and this is given here. So you can clearly see that we have transformed some cells, and then he made colors and got regenerants, and then he analyzed those, and the outcome is given here. So this is the target sequence. And amongst 231 individual mutant plants which he obtained, so he made pools of 8 plants in order to reduce the number of samples he had to send. He gave a unique barcode that later on we can still understand which at least batch of plants the reads are referring to, and sent this to next generation sequencing. And the predominant outcome is given here on top. So it's a -3 deletion in the correct site. And if you are wondering why we got this -3, so this is because here we have this triplet twice. So GCT occurs here twice, and maybe even the cell thought that it had correctly repaired this double strand break. And the good news here is that 25 of those pools out of 38 contain such deletion. So it shows that this system can be used in order to manipulate barley plants. And there are examples of other groups which also have published that they can use RMPs in plants in order to target their genes. Another one, as I briefly mentioned, is that our primary regenerants are usually chimeric. And in order to solve this problem and to fix our mutations in cereals like barley, we can apply some double tabloid technology, which is based on this embryogenic pollen culture. This means you harvest immature spikes of your primary regenerants and start an embryogenic pollen culture. And then after some induction, you end up with some plants which then are no longer chimeric and are homozygous for your mutation. And my former colleague Dia Dagmar has looked up for this process and did here this nice cartoon. So under certain stress treatments, you can render a pollen grain, which is usually going for this normal pollen development. You can alter this in order to start embryogenesis. And so we have analyzed some of the treatments which are necessary in order to switch from normal pollen growth into this pollen embryogenesis. And so what's going on here? So if you have your primary mutant with your mutation in one chromosome, then into your pollen, of course, you spread all your different parts of the genome. And then after genome duplication, you get your DH plant, which is homozygous for your mutation. And to give you also one example here for the same target, this LOX1 gene. So here in this case, we used 2 different guide RNAs targeting 2 different positions adjacent to each other. Again, in red, the target site, and in blue, the PAM. And in gray, you can see here the primary mutant, which consists just of this -3 deletion here in this one target site. And then if you look in the progeny, the DH progeny out of this plant, then you can see that, yes, in all those DH plants, you still have this D3 mutation, but you detect also plants and individuals which have also additional mutations in the second target, which was not present in the sample which we analyzed in T0. And this is true for other ones as well. And here I also want to highlight that you can also get plants, homozygous plants, which then has also a deletion between 2 target sites, which would even give a predictable outcome. Because usually what we get in this targeted mutagenesis is still random. We can't predict if it would be a -1 or +1. But by using these 2 targets in a certain number of plants, you can predict the outcome, which is another advantage of this technology. Another way to circumvent transgenesis has been published also by my colleague Nagaveni Buddhakattapalli. So she used white hybridizations in order to induce mutations in the plant of interest. And so here I give you an example for corn and wheat. So as we are very efficient in making transgenic corn, but wheat is more resistant for transformation. And you will later hear, of course, nice success stories of Sadir and Mark, how to circumvent this and have efficient protocols. So we choose a different approach. So we made the transgenic maize plant, which express the guide RNA and the Cas for the target in the wheat genome. And then you pollinate your zygote of your wheat zygote with those transgenic maize pollen. And after fertilization, you usually get rid of your corn genome. So this will be cut out of during the further cell divisions, and you end up with a maize plant which harbor just the mutation and is not transgenic at all. Sorry, I think I told maize plant. Of course, it's a wheat plant, which then has the mutation, and the corn genome becomes eliminated. And that this is true is given here. So she used bread wheat and durum wheat to test this hypothesis by targeting the BRY1 gene. And so here, as you can see, we used also different genotypes. Bob white is the most common bread wheat spring genotype used for transformation. And she also used different genotypes for bread wheat and for durum wheat. And here you can see also the target sites. And in green then is highlighted the modifications she could find in those plants. So the approach is working and has been published. So please go for the publication if you are interested in more details. Another problem heavily discussed is also off targets. Also, I think in plants, this is not that much a serious problem, as if a plant looks weird, of course, you would throw it away and you would not continue growing it. But just to make a serious science, so here I will give you one example where we analyzed, again, it's still on the same target, this LOX1 gene, which has the on target on the arm of chromosome 4H. And most likely due to genome duplication events, we have another putative off-target site nearby, also on chromosome 4H, and another putative off-target site on chromosome 7H. And here, Punepuramini also checked for those off-targets by amplifying the respective targets. And here I will give you first the overview for chromosome 4H. So first, of course, you should look that your plants have in your desired position a mutation, and this is given here. This is our on target. This is the sequence of our wild type. And then here in the plant, you see a -1 deletion. And then, of course, in the same plant, you can look if you find also mutations in the off-target nearby, and she did. So, and this is given here. So as I say, there are putative off-targets, and putative means that there might be some single nucleotide polymorphisms within the target sequence. And here in this particular example, you can see that there are already 4 different ones. And as you can see, none of the sequences show alterations beside these SNPs, which are also present in the wild type. And then looking for the other target on chromosome 7H, the same happens as well. So if beside the single nucleotide polymorphisms, there are no other modifications in those plants. So also there are publications showing that also off targets can occur. I do not neglect this, but of course, one should always check, and one can prevent having off targets in careful selection of the guide RNAs. And I think the next talk will also guide you how to select good guide RNAs. So last but not least, so some words about the legislation, as this was also already in the discussion. So the effort for the approval of a genetically modified plant, as also in Europe, those gene-edited plants are treated at the moment, are estimated for roughly $130 million. And it takes nearly 13 years to come from the work in the lab until you have a commercial product. So which is a lot of money and a very long way. Because where a lot of authorities are controlling this, but there is also a lot of science indicating that there is no additional risk out of those plants compared to our breeding methods. But there are, of course, outside of Europe, GMOs on the market, and some of them are listed here. So they're quite a nice number, reaching from cotton, corn, soy, canola, sugar beet, alfalfa, squash, and so on. So there are those products on the market, but these are not mainly gene-edited plants. So these are herbicide-resistant or otherwise transgenic plants. And here in this overview, you can see that quite some part of the world are growing transgenic or GMO plants for the benefit of the public. If you look for CRISPR technology, so then it becomes more weird, because as you can see here, there's still a lot of green. So a lot of countries in the world do consider gene-edited plants differently than Europe. Also, one should differentiate the outcome. Of course, gene editing can make just small modifications like single nucleotide polymorphisms or small deletions and insertions, indels to be precise. Yeah, so you can make small modifications, but of course, by using the homology directed repair, you can also insert foreign genes or can even modify a plant in a different way. So the technology allows more than this. And one should differentiate in the application and if a plant is considered GMO or not, depending on what have been done. As long as you introduce foreign DNA or assemble a plant in a way which is naturally not possible, one should still consider it as a GMO, but at least if you have small modifications which are not distinguishable from what happens in nature or what has been introduced by, for example, mutation breeding using radiation or EMS, so like cytotoxic chemicals, so should not consider GMO. But as you can see here, the regulations are quite diverse, and so it's hard to grow these plants in Europe. So at the moment, it's not forbidden, the politicians say, but it's a very long process. But there is some hope. So because the European Commission is now in the process of reconsidering to modify the present GMO rules, so there is some hope at the end, but the authorities are of course very slow, so I do not expect some major changes before the year 2024. But there are also gene editing plants on the market. So the most prominent and first one might be the cibus canola, which you might be aware of. So which is based on a oligonucleotide directed mutagenesis. Then there is also now, and as also Mark outlined, so with using gene editing technology, we of course now in a rather simple way can bring a wild plant into a crop plant. as has illustrated for tomato, as given here, the example, by just altering 4 or 5 traits, you can come from a very unproductive wild plant into a very productive crop plant. And this has been also illustrated for wild rice. So, and more of those applications for sure are in the pipeline. And so, as also mentioned before, so tomato is one of the products which also reached the market. There is this GABA tomato, which has been released for the market in Japan. And this has some health promoting benefits, eating this. And there is another one, which is in the pipeline of Syngenta in China, where we want to prolong shelf life. Also, this has been done also previously by other genetic modifications. And last but not least, so also Mark mentioned this, that we are run off agricultural land, and also Mark provided nice examples that there are already salt-tolerant plants which can be either converted into crop plants or introgressed. So we can use all the breeder's toolbox in order to make use out of this. And there's also one company in Canada, Agrisea, which is also at the moment developing floating ocean platforms in order to grow plants on saltwater, so on oceanic saltwater. So Mark mentioned using brackish water for irrigation. Here it would be even going more far to make those farms on the sea, as of course there is a lot more space on sea, and maybe here in these coastal areas we can expand agriculture to this if we are able to turn crop plants in a way that they become salt resistant. So there are things on the way using gene editing in order to reach the market. Either they did already or they're close to. And with this, I want to close. So I want to acknowledge the work I have been done in the former group of Jochen Kumlin at IPK. I want to thank my funding, and I thank you for your attention, and of course would be happy to answer any question.
Dear Dr. Hansel, thank you very much for your presentation. Now I would like to take a few questions from the physical audience and maybe one question from the online participants.
I just wanted to know that this is our experience that the efficiency of genome editing showed your R&Ds, if I can remember, it says that out of 38 embryos you got editing in 25.
I'm sorry, it seems there's a problem with my internet connection, so I couldn't hear the questions. I'm sorry. Can you please repeat the question? Hello? I'm sorry, I could not hear. Sorry.
Can you hear me now?
Yes, now it's better. Yes.
Okay. So our understanding and experience is that the genome editing efficiency is a little bit poor. When you showed the results of your R&Ds, and you said that if I'm right, that out of 38 embryos you got editing in 25 embryos. So would you like to elaborate that? This is a really high efficiency.
Yes, so I was also a bit surprised to have this high number, which might be also related to the target site. So as I showed you, so in this location, this might be a position which is highly amenable for inducing a mutation. So for whatever reason, yeah. So we have in stable transgenic plants using agrobacterium-mediated transformation in protoplasts, and also by using the RNPs, we always ended up with high efficiency. So that's why this guide seems to be really very efficient. And I would not say that every other approach would be as efficient as this. So here, this might be a special case. But what I wanted to show with this, and others have shown as well, the RNP technology is quite useful in order to avoid also that you want to get rid of your transgene afterwards, after your mutation is there. Yeah, so because then usually the job is done and you want to get rid of the transgene, not because of the public concern, just to be sure that you have a stable genetic plant. Yeah, so that's why usually we segregate out the tDNA if we use tDNAs. And so in this regard, using the RNPs, of course, can speed up this process, especially in crop plants where you save 6 months to 1 year, which is, of course, quite substantial, not just for PhD students.
All right. Thank you very much.
Thanks for the question.
Now we will take one more question.
Hi, Dr. Hansel. My name is Musa Kavas. I am from Monocots University in Turkey. My group also working on the genome editing of tomatoes. So we successfully create some genome edited tomato lines. Especially we create some male sterile tomato plants. So during our experiments, so according to my experience, so we couldn't observe any differences efficiency when targeting positive strand or negative strand. But recently we started a new project, so we are trying to overexpress some tomato genes. So, but when we check the literature, so we observed that there is a difference between during the guide RNA design. So there's a difference between when targeting positive strand or negative strand. So, for example, if you target negative strand during the overexpression of by using that Cas9, so there is a highly difference. So, according to your experience, did you observe any difference when you use normal guide RNA or a normal knockout during the CRISPR Cas9 or the overexpression? Did you observe any difference during the guide RNA design?
Many thanks for this question. So, of course, over the years, we accumulated a number of features in order to identify efficient guide RNAs. And most are covered also by the online tools which are available. And according to my experience, even from different plant species, targeting the sense or the antisense strand made no difference in the experiments we have done. So we also looked carefully if we see differences in this direction, but it's rather the secondary structure, the nucleotide composition, which nucleotide is in which position, and so on and so forth, and maybe accessibility of the DNA itself. So where is the chromosomal location of the target, which influence your targeting efficiency than the plus and the minus strand. So I did not see a preference for this in our experiments.
And did you study with overexpression with CRISPR?
For the, yeah, so what we have done so far on homology directed repair. So we have done a switch from GFP to YFP, as these are just different in 2 nucleotides. And here we were targeting the plus and the minus strand, and I also did not see a difference in the efficiency, but I have to admit that this is an artificial target. Yeah, so this is, of course, it's a transgenic plant constitutively expressing GFP, and this was targeted by endonuclease, and we provided a non-functional YFP fragment, which has been used for the repair mechanism, and we find a quite high number of cells which do have this allele exchange. But unfortunately, as this does not give selection benefit, so also screening 300, 400 regenerants, we was not able to got one of those allele exchange plants. So my results are mainly based on either biolistic experiments in epidermal leaf cells or on protoplasts. So I can't say anything from own experiments with stable plants in this direction.
Thank you.
Thanks for the question.
Okay, thank you very much. Do we have, then we can actually take.
I can ask this. I'm trying to, there's no question that I speak.
Okay, so about the gene editing and bioethics. So the thing about the editing and its possible effects on the bioethics.
Can you shortly repeat, because there was again some connection problems? So,
Okay,
and what do you mean with the bioethics?
So this was a question from Ms. Zeynep from the online. So if Ms. Zeynep, can you just ask your question yourself, if it is possible? Maybe. So I think the question is looking for the ethical part of the gene editing in crops. So can you give some brief information about it, what you think about that?
So, yeah, bioethical point. So I'm not an expert in bioacid ethics, so that's why my comments might be not that well educated. So I think depending on the outcome or on the approach you are doing, so the gene editing gives us for the first time tools in our hand where we can modify a plant in a specific way. And as farmers and breeders are doing these gene modifications in a more random way since centuries, so I do not see a point in a specific point which can be raised here, rather than being more specific should not be a problem. Yeah, so if we can do one thing better, so this should be not a point of complaint. So, but I'm not sure that this answer the question here. So I think with these tools, we are now able to do very specific and precise modifications. And this was also my hope, that by showing the precision and what minor the modifications are compared to classical breeding methods, that we can also convince the public, which is usually against because of misinformation, so that we can convince them better. So, but I'm not sure if this was the ground of the question and if this answered the question.
I think it explained it very well. Thank you so much. So we had received one more question from the online audience. Actually, two questions. One question is that if we can use this technology in linked genes, this is the first question. And then the second one, that if we can apply this technology to woody plants.
Yes, so I think in the table was also mentioned, and I am aware of colleagues in the Thunen Institute, which are also working on poplar and doing gene editing in poplar, and there are others, I think, also working in other woody plants doing gene editing. And of course, the generation times are much longer, but it's also possible. And for targeting linked genes, I think this is also ideal technology. So because the group of Holger Puchter had also showed that they can now solve one chromosomal segment which was so far not accessible by recombination, that they can also now use gene editing technology in order to force this further, so that this will also be possible, because usually in the typical crossings, linked genes can't be separated. But now by inducing double strand breaks and maybe making some chromosomal rearrangements, then you can even bring this further. Yeah, so this will be possible by using gene editing technology. Yes.
Okay, great. Now we have one last question
You're
from
welcome.
Dr. Allah Baksh.
Hello.
Hi, nice to see you.
Good, thank you. Continuing from Dr. Imran's questions, yes, that's right. Editing efficiencies in RNPs is better than plasmids. Now the question is, can all plasmids vectors, can they be delivered in RNP format? This is one. Other, the same Cas9 enzyme that works well in RNP, what about its efficiency in form of plasmids?
So,
I mean, did you try it?
We have used, so the postdoc, I showed one of the results for the RNPs, Martin Becker. So he compared on one hand different Cas enzymes, the Cas12a and the Cas9, the SP-Cas9, for the efficiency based on our stable transgenics. We had ideas how the same enzyme is working on a plasmid if we transfer it as a plasmid, and he also used the same enzyme as ordered from a company for using the RMP, and we did not see a difference in this. So I have to admit that Holger Puchter has published that different Cas enzymes, depending on the source, this SA Cas9 was much more efficient in Arabidopsis than the SP Cas9. So depending on the plant species you are working with, so of course it's wise to try or not to stick just to the most common Cas9 protein. So you may achieve much better efficiencies by using different Cas proteins, and there is a high number. So because this has also the advantage that also I could not go into the details, that each Cas protein of course have different requirements for this photospacer adjacent motif for this Pam sequence, and so therefore these different Cas enzymes make it more flexible amongst the genome to go for your target sites, because with the NGG of the Cas9 you are of course limited in selecting your targets. But we have compared plasmids and RMPs, and we, at least in our experiments, we did not see a difference in using plasmid or the RNP for the same protein. But as mentioned, by using different proteins, you see differences. I hope this answers your question.
Thank you for elaboration. Thank you.
You're welcome.
Okay, thank you very much, Dr. Hansel, for this great talk and this intensive lecture about the genome editing in plants. With this, I would also like to thank everyone for joining us virtually as well as physically. We will give 1.5 hours of break, and then we will continue with the rest of the talks. Thank you very much.
Many thanks. And enjoy the remaining workshop. So I wish you good success.
Thank you so much.
Okay, Bismillahirrahmanirrahim. I think that it's a pleasure to welcome you in the afternoon session, and I believe that it took longer than expected. So, but finally we are here, and our colleagues online are also waiting. So, may I introduce Dr. Imran? Is working as principal scientist at the National Institute of Biotechnology and Genetic Engineering. He's I've been working with me right from the beginning, when he was an MSc student. So he's a very close and long association. Dr. Imran Amin basically got his PhD degree in characterization of viruses and suppressor of gene silencing encoded by some of these plant viruses, specifically plant germ viruses. And so one of the approach that we have is to have gene silencing, and then when we so we use different technologies. I myself work with Professor Welcome on mRNA technologies, but now we have developed these new technologies for silencing or mutating genes by using genome editing technologies. So we will talk about this topic, which is more related, about designing how these tools. Besides designing tools, we also work on different versions of Cas proteins and their applications, and you might have seen publications coming up from our lab, basically to develop new tools. Thank you.
How to share? Can you? Good morning, ladies and gentlemen. Thank you very much, Dr. Shahid, for introduction.
Just share screen, and this.
So I've been asked to sit here because we have virtual participants as well. It should work now? No. Understand this.
Okay.
Presentation mode. Do we have a presenter?
We don't have a presenter. Can you use the one?
We don't have this. Stop share. Share whole computer. And
okay, it's sharing.
Share this one. for this delay. And then secondly, what I think is that it should be a practical session rather than a lecture, but unfortunately we were unable to manage that. So I'll try to manage it with some screenshots. I will share some web pages. I'm here for 3 days, and if anyone is interested, we can work on the computer and we can work on their guide RNA. So the content, this is not a research topic, it's basic. So I'll discuss what is genome editing, of genome editing, because that is required for designing your guide RNA. Then a brief history of CRISPR-Cas system, and then toward the end, we will discuss in silico guide RNA, designing for CRISPR-Cas system. So if you talk about CRISPR, it's a bacterial immunity system, first discovered way back in late 87, 88. It is working against bacteriophages. It is a very conserved mechanism. It is found in Photosynthetase and about 90% of archaea. Is that is a interspersed syndromic repeats. It is the CRISPR is based direct nucleotide repair, which are separated by the and then there is a protein associated protein which use those guide RNA as a it results in the cleavage of the DNA. Naturally, what happened is that when bacteriophages infect a cell, that it will inject that cell. That DNA like this. If this is the DNA, it is recruited into the CRISPR array. So what it does is that whenever there is a secondary infection of that, this DNA is acting as a guide. It will, and that RNA will guide this Cas protein, and it will cleave that bacteriophages. So there is no secondary infection, theoretically speaking. How we use this system in I have given the example of the plant is like that we develop our own guide RNA. We develop, I'm talking about, we develop the construct of Cas9 as well. And then we transform that into the host cell. In the host cell, the guide RNA is and then this Cas9 is produced. This guide RNA will guide this Cas9 protein, and it will, of course, there is a sequence which is called as Pam. I'll discuss this in detail in the later slide, that it will result in the cleavage of the DNA. Okay, another thing is that when we talk about the repair of the DNA, the cell has this natural ability that it can repair itself. There are two kinds of non-homologous result in the indels, we call it insertion and deletion, and then there is homology-directed DNA is the knockout. So this genome editing is a technique where mutation, you can also produce the knockout gene. So it can be used in the synthetic biology, it is used in gene therapy, and there are certain Cas proteins which are used for the gene regulation as well. If you see that in my career, observed is that this is one of the technology that got price in a very little time. The first one, if you remember, that was the Caramulus, the PCR, and then the second in biological sciences is this. That this era of we divided into three. The first is identification phase, and then the characterization, and then its application. The applications are huge, not only in plant sciences, it is in the animal sciences as well. Now I'll discuss very basic, which is required for designing the about CRISPR-Cas9. Let me first tell you that there are around 90 Cas proteins, and each Cas it has its own cutting mechanism, how it cleaves. Its requirement is different, so you have to take care that which Cas protein you are talking about, design accordingly. The most widely used protein is Cas protein is Cas9 protein, which I mean most of the time we work with the Cas protein. The class 2 type 2 system. This is very important. This is protospacer adjacent motifs. We call it Pam sequence. The Cas protein require a Pam sequence to cut down or to be guided by the guide RNA. In case of Cas9, this is NGG, but for every Cas protein, Pam sequence is different. Then there is a sequence we call the seed sequence. If you see here, this is basically the guide RNA. The guide RNA size of the Cas protein is 20 nucleotide. The seed sequence is, this is Pam sequence, and this is the guide RNA. That's proximal sequence which from this Pam sequence up to 11 nucleotide is basically the seed sequence. Seed sequence is very important. If you remember the previous lecture where Dr. Hensel was telling you about the off-target guide RNA. This sequence is important because if there are mismatches in this sequence, Cas protein can tolerate that. If there are mismatches proximal to the seed sequence, Cas protein will definitely cleave that and it work on, it will produce its effect, what off-target effect. Then there is a CRISPR RNA. If you see, this is the guide RNA. Upstream of the guide RNA, there is another RNA which is called as CRISPR RNA. This CRISPR RNA basically bind with the tracer RNA. A functional guide RNA constitute the guide RNA as well as the CRISPR RNA. CRISPR RNA bind with this tracer and it form an arm that on which Cas9 detect not only the PAM, but it also detect this tracer RNA and it act on that, and then it result in the cleavage or whatever function we wanted to have. I'll first tell you different Cas system on which we are working, and then I'll discuss about the designing of RNA. This is CRISPR-Cas12a, we call it CPF1. It is class 2 type 5 system. The size of the CRISPR protein is around 1300 nucleotide. And by the way, Cas9 is also 1300 nucleotide. It is capable of processing its own CRISPR RNA, and it uses only ROV-C domain. It's basically the domain that is responsible for the cleavage of the DNA. Cas12 does not require any tracer RNA, so this is different from the Cas9, that there is no need of tracer RNA for that. And the size of the guide RNA is around 41 to 44 nucleotide. In case of Cas9, it was 20 nucleotide. In case of this Cas, it is 44 nucleotide. The PAM sequence is basically 5' TTN, which is, N is for any nucleotide, which is located directly upstream. The main difference between Cas12 and Cas9, sorry, if you see this, the guide RNA is on downstream. It is on the 3' end. In this case, it is on the 5' end. So every Cas protein has its own guide RNA, the size, and its direction, whether it is present on the 5' end or it's present on the 3' end. Another property of this CRISPR-Cas12 protein is that the indiscriminate cleavage of collateral single-stranded DNA. Once it is activated by the guide RNA, it worked on the double-stranded RNA. But when it is activated, it indiscriminately cleaves the single-stranded DNA. And this is very important in a way because there are certain viruses which are single-stranded, and we cannot target single-stranded viruses. Though we have another Cas protein that can target that, but it can target double-stranded RNA. It can also target the single-stranded RNA. This is another system of the CRISPR, which is CRISPR-Cas13a. This is CRISPR. Previously it was known as C2C2, and it is class 2 type 6 system. It is an RNA-guided RNA editing system. The previous two Cas proteins which I was discussing, those basically cleave the DNA. This CRISPR-Cas13a, it cleaves the RNA. So we have both the proteins that can cleave the DNA as well as the RNA, and Cas13a is the one that can cleave single-stranded RNA. It has dual function of CRISPR-Cas13a system. Pre-CRISPR RNAs are processed into the mature, so its CRISPR RNA is produced by itself. The size of its guide RNA is around 28 nucleotide. This is another system, which is CRISPR-Cas14a. This is relatively a smaller protein of about 400 to 700 amino acids. The size is 40 to 70 kDa. Another property of this CRISPR or Cas protein is that it don't require Pam sequence. So if you give it a guide RNA, it will result in the cleavage. And the cleavage is basically of the single-stranded DNA. It don't restrict or it don't cut the double-stranded DNA. And the guide RNA size is 37 nucleotide. Now let's talk about up here. The point which I want to make is that we have different kind of Cas protein. You need to know which Cas protein you are working with, what is the requirement, what is the Pam sequence, and then whether that Pam is present upstream or it is present on the downstream, and then you design your guide RNA. If you talk about the summary, it's a whole of the genome editing. That first thing is you need to know the sequence. Mostly either you download that sequence from any sequence database services, but we prefer to have sequence of the crop or the plant which we are working on, so that you have exact same sequence and you can design the guide RNA. Then there are certain software which I'll let you know, but I'll tell you there are several the software which we use during this, and we also, by the way, I'll also tell you how we manually design the guide RNA. When those guide RNAs are designed, one of the most important part is to know that whether they are going to produce off target or not. You know, the size of these guide RNA is around 20 nucleotide or 40 nucleotide, and this homology, you can find it anywhere. In humans, in plant, this is less problem, but when you talk about the mammals or the higher animal, it is a problem. So you need to you design your guide RNA in such a way that they should not produce the off-target effect. Once you get that off-target, you find that good guide RNA, you clone it. We use Golden Gate cloning in that, and then you transform it, and then you do the sequencing. I think the previous lectures has shown you that how we do that. So guide RNA for the Cas9, for the example, I'll show you the Cas9. First of all, if you see, this is the Pam sequence, and the guide RNA is the 20 nucleotide which is present downstream toward the 5' end of this. So this will be the guide RNA, and this is the Pam sequence. The Pam sequence itself is absolutely required for cleavage, but it is not part of the guide RNA. The Pam is not included in the guide RNA. We design guide RNA without the Pam sequence. If you're talking about mismatches, so the limit is 3 nucleotide. If you have 3 mismatches in your seed sequence, as I mentioned earlier, the guide RNA will tolerate that. The Cas9 will tolerate that, and it won't go for cleavage. But if that homology is, the mismatch is away from the seed sequence, then it will definitely produce the off-target, and you have to take care of that. I'll show you with few examples later. These are the different guide RNA designing tool. As far as their work is concerned, it's simple search. What you need is the Pam sequence, even in the work. So you can design the guide RNA. These are, I'll go through the, I'll show you screenshots. For example, this is GeneScript, and if you see that these are the genomes which are available on this website. These genomes are basically the reference to tell you the off-target, because when you give your input in this in the server or in this web page, they will end up with the guide RNA, and they have at the back end they have these genomes, and these genomes, they will tell you that whether your guide RNA will produce off-target effect or not. This is basically Eukaryotic pathogen CRISPR guide RNA design tool, and there is a lot of flexibility in this. I usually prefer this for designing the guide RNA. What we do is that if you see here, first you have to tell this this server or this interface that what is the PAM sequence, what sort of Cas protein you are working with. Here, these are the different Cas proteins. Each Cas protein has its own PAM sequence. So you first have to decide that which PAM you require, which Cas protein you are working with, and then they have. If you see here, they have different genomes. You can, out of these genomes, you can identify or you can select the sequence, or if you go there, you can also give your custom genome. By custom genome means the sequence which you will give, which you are interested in, and you can have the guide RNA. It will give you an output like this. For example, this is a cotton leaf Cochrane virus. I just wanted to know that where are the guide RNA. If you see that, it will give you this. This is the PAM sequence. Of course, this is not part of your input, but it gives you that this is the PAM sequence. It will give you the score. The score it gives basically that they have come up with the numbering, 1 being maximum and 0.01 is the lowest, where it numbers, it gives you a score based on the specificity of that guide RNA and its chances to result in off-target effect. It will also tell you that during transcription, what could be the problem and what nucleotide you may have to add so that you can have a proper RNA in that. This is another web page where a web tool which you can use, but it is only for these organisms. But if you see here's Arabidopsis thaliana, you can use it for Arabidopsis thaliana as well. This is specifically for the stem cell, is for a human and mouse genome, where it can work with. Okay, this tool is for CRISPR cereals. There they have wheat, they have rice, both Japonicum and the other one, and then Xiames. So if you want to work on that, you give your input in this, and it will provide you your guide RNA. This is another tool because it has flexibility again. Here you can, they have their genome as well, and then you can also give the custom genome which you want to work with. This is called CRISPR Direct. If you give your input and you have to select what sort of PAM sequence you are looking for, and then based on that PAM sequence, it will give you the guide RNA. If you see here, this was a custom genome, means the genome which I want to work with, it cannot give you the mismatches because that genome was not part of this web page. This is another example where I have given that I want to see that how many mismatches are there in case of human beings, and the same output comes with this, and it can tell you that how many mismatches are there and what are their position, whether they are present in the seed sequence or they are present away from the sequence, from the seed sequence it can tolerate or not. So the thing is that you have to identify the best guide RNA that will suit you. This is another tool which is called CRISPR P2. This is again very widely used. You give your input in that, and it will provide you the guide RNA along with the PAM sequence, and then it will tell you the GC ratio and the other stuff. Now, up till now, these are the different other tools which you can use. Designing is not very difficult. It is basically the selection of the guide RNA, which is the best guide RNA that can be utilized. That is done by if you are sure that it don't have off-target effect. Before that, let me tell you that how we do manually. Manually is very simple, as I mentioned earlier, that you need to know what is the PAM sequence, where your guide RNA lies upstream of the DNA or downstream of the DNA, how many nucleotides are there, and then you can take out that sequence and then see that whether this is going to work, it has some off-target effect or not. This is one of the example, one of our student, she was working on potato. It is basically to develop resistance against potato. She was working on ELF. It is an elongation factor in the, I'm sorry, potato. It was ELF4 is is an elongation factor with which TVY, potato virus Y, interact. So if you stop that interaction by mutating the ELF4 gene, there is a chance that you get resistance against those viruses because that virus cannot replicate. So what we did was that we not only downloaded, we also sequenced that. We find the ID of that sequence. And then another thing is that usually you design your guide RNA not on the intron region. It has to be an exon. So you identify the exon region where the exon lies. After those, there are several software. You can do it in any software which you are working with. We highlight those exons, and from those exons, you identify or you find the Pam sequence. And if you're working with the Cas9, you go 20 nucleotide downstream of that sequence as a guide RNA for you. As far as cloning is concerned, really, these are oligos which we synthesize, and we use type 2 restriction endonuclease golden gate cloning, in which it produces 4 nucleotide overhang. We add those overhangs with our guide RNAs so that it can be cloned in those particular vector. And I'm not going to detail because the next lecture is about those the vector system, and Dr. Zahid probably will tell you about how we do cloning in that. Now, this is the most important part. You have designed, you have the guide RNA, you can have hundreds and hundreds of guide RNA because these Pam sequences, they are NGG sequence. But the problem is guide RNA, and that guide RNA should be the one that you, and in this case, I mean, blast, everyone probably must have done the blast. You can do blast in batches, and you can do 100 of those nucleotides. You can blast homology, or you can restrict that blast to a certain particular organism. You want to seed off, you can do that blast as. We use several tools. One of the is this this cat. I can web pages with you. What you have to do is that your input here, the guide RNA, the 1000 guide in one go. So what you do is that Excel file here, and you how many mismatches, up to 3. You have to put, because of that, so once that the organism which you are working with. For example, what I did was Marsutan, and that. Anyways, if you see as an example, this that is giving you effect of target of 1 number, and this is a guide RNA that there are 253 off targets. So obviously you will select the one which has minimum number of off target. For example, this is the detail of that experiment. If you see, this is the seed sequence. I told you that it is about 12 nucleotide. If there is a mismatch here, this can be tolerated. Ideally speaking, there should be no mismatch. And there are certain guide RNAs. It shows only those guide RNAs that shows the mismatches. Those guide RNA you have to work on a particular sequence, so you don't have this liberty. In that case, you have to see that where that mismatch is, whether it's in the seed sequence. The thing is that it should be in the seed sequence. You can go with that, but if it is not in the seed sequence, then you cannot use that. You have to come up with some other strategies. This is the cloning. That is all. This is basically what I want to show here is that these are the nucleotide overhang. This is the guide RNA to here, 20 nucleotide, and the overhang are introduced just to clone them in the golden gate. Golden Gate vector. This is in silico cloning, which we usually do once we design guide RNA. There are certain software which can do that, and that is all from for the designing. I would like to acknowledge COMSTECH, COMSEC, and me too, and thank you very much.
Now, I think we should take the all the questions at the end of the 3 sessions. That would be better. Okay. So we will continue with the next speaker. This next lecture will talk. We will have the lecturer from Saudi Arabia, Dr. Zahir Ali. Dr. Ali works on genome editing in different crop species, and today he is virtually joining us on this Zoom, and he will talk about the vectors used in the genome editing, and basically he will tell us how to design those vectors. He should be here.
I think he is there. Yeah.
Okay.
Okay, Shamshad, please unmute Dr. Zahir Ali so that he can speak and share his video and slides.
Hello.
Okay, yeah, go ahead, Dr. Zahir. Please turn on your camera as well. We cannot see you. Yeah, now.
Can you see?
Yes,
Yep.
yes. Please also
Yep.
share your slides.
Can you see my slides?
Yes, we can see.
Yep. Yep. Thank you for the nice introduction. I'm Zahir Ali. I'm working as a research scientist at the King Abdullah University of Science and Technology, Saudi Arabia. Basically working with the plant genome engineering and specifically with the CRISPR-Cas system, which is now converting mostly into every field, including genome editing plus diagnostics. Today, I will present some work about the vectors used in genome editing in crops. So I will talk about different vectors that are used in the plant system. Before that, I will talk about as Dr. Imran already presented this one, but I will just briefly introduce that the CRISPR-Cas system is basically the immune system of the viruses, the immune system of the virus, and basically it's a memory system, like when the virus attacks the bacterial genome, the bacterial cell, and the genome of the virus enter into the bacterial cell. The bacteria keep a small piece of this virus, the virus part with itself in the genome as a memory. And this is called like immunization. So now the bacterial, if the bacteria have a small sequence from the virus genome as a spacer, then it can give memory for the next attack. So like, for example, when there is the virus DNA again, or the virus attack, the virus come back to this bacteria, this gRNA or this spacer is expressed in the form of sgRNA, and this make a complex with the Cas9, and then it can target the virus genome. And then after this targeting, the virus genome is destroyed. So this is the immune system of bacteria and archaeas. And very specifically, they are present for the RNA viruses or the RNA phages or the DNA phages. And the system is very simple. like it's need a simple Cas9 or the Cas nucleases, and it's need a guide sequence that can go to target DNA. So looking at the system, looking at the simplicity of the system, when these systems were discovered, it's very easy that it can be used for genome editing of any DNA. So like, for example, it's just need a very simple sequence, like you need only 20 nucleotide sequence, followed by an NGG or a Pam sequence, which is different for every Cas9 nuclease. And then you can put this into a gRNA sequence, and here is a scaffold which is binding, which is responsible for the binding with the Cas9 protein. So when these two combine, it can come to the target DNA, and then it can make a double strand cut, because here we have the rough pseudomain and here we have the H and H domain, which make two cuts on the DNA, and the double strand cut is there. So these are the basic requirements for the CRISPR-Cas system for the genome engineering or genome targeting. But then normally how we are using this system and how we can clone this system, normally we are using, there are different methods like the restriction ligation system, the gateway system, and nowadays, because the CRISPR-Cas system, it's very it's need very a lot of cloning and a lot of gRNAs we have to check. So it's need a simple method to clone the things. And one of the simplest methods recently people are using is the Gibson assembly, which is very straightforward. You just need a vector that even you can cut with the restriction enzyme, or you can do the PCR for it. And then whatever you are cloning, like for example, if you are cloning the Cas9, we need a tag, we need a nuclear localization signal, and then you can just amplify, PCR amplify with a specific sequence overlap, and then you just mix these three things, and then in a single enzyme and at a single temperature at 50 degrees C, 15 to 60 minutes, you can clone all the fragments. Like for example, you can go up to 12 fragments, and even the whole genome of bacteria can be cloned with this method. Just the PCR fragments are there, and then you can make them into a single clone. How it's work? There are three enzymes basically working in this system. One is the 5' exonuclease, which start cutting from the 5' end. And then we have a DNA polymerase, which fill the gaps. Like for example, these two overhangs are there. So the 5' end, it start to degrade this one, and here it start to degrade this one. So then you have a complementary sequence for one another. The same is happening here. And then a gap is there because the exonuclease remove some sequence. So when the gap is there, this gap is filled by the DNA polymerase. And then the ligase. So when the gap is filled, then we need a ligation. So here the ligase will come and it will make the clone. And here the same will happen. And these three enzymes work together at this simple one temperature and one tube. And then you just transform this into bacteria, and you can see the colonies transformation very easily. Compared to the other system, this system is very simple and it's very easy and very robust. Like for example, you can see almost 99% of the colonies, they are positive, and it's very simple compared to the other methods. But this is just about the cloning. All the clonings can be done with this simple method, even though the plant vectors, sometimes they are a little bit big. But even if the vector is big, you can just do a pieces PCR. Like for example, this, if we are amplifying this fragment, so we can do a PCR here, then a PCR here, and then a PCR here, and then we have 5 fragments. So all the 5 fragments can be put in single tube, mixed with the Gibson assembly, and then transform. So you will see a whole clone and then the transformation. So the system is very simple. Then what are the basic requirements for the CRISPR-Cas system for plants? The first thing, the CRISPR-Cas is a bacterial system, which is a prokaryotic system. And it cannot be expressed as it is because it's a very big proteins normally. Nowadays, there are some small proteins discovered in the CRISPR-Cas system, but normally they are big protein. And this much big protein from a prokaryotic system are not able to be expressed in the plant system or in the eukaryotic. So the first thing is like codon optimization, because the expression is basically dependent on when the RNA is there, then we need the codon and anti-codon system to make the amino acid or the polypeptide chain or the case nucleases. So the first thing is like we need a codon optimized case nuclease. Whatever, like for example, if we are using Cas9 or if we are using Cas12 or Cas13, whatever, but for the eukaryote, we need the codon optimization. This is the first requirement. The other thing, like there are different case nucleases. So which one we want to use? And they have different advantages and disadvantages. Like for example, Cas9 is very robust, but it have some off-target effects. Cas12, it's also working very well. There are a lot different available. So whatever we are using, we have to select very precisely for our purpose. The second thing is like all the case nucleases, they don't have any NLS. And the genome, compared to the prokaryotes, the genome of the eukaryotes are in the nucleus. So we need to put the NLS tag with it. And if we want to detect by the Western blot or by different methods, so we need the other tagging like flag tag or the HA tag. We also need these things to be in these sequences. And then the orientation, as Dr. Imran explained in the previous lecture, that the orientation of the guide sequence is very important. And the Pam sequence is basically determining the orientation, like where is the Pam, and after that the guide sequence will be there. So it's very important to select a very precise orientation of the guide sequence. Then selection of the nuclear expression promoters. This is the most important one because all the genome is there in the nucleus, and specifically the gRNA, like the Cas9, we can put the NLS. But for gRNA, it must be in the nucleus. So if it is expressed from a promoter, which will take it out of the nucleus, like for example, the pole 2 promoter, all the RNA is expressed from it, it's go out. So we need a pole 3 promoter to express the gRNA to keep it inside the nucleus. And when the NLS with the Cas9 is there, and then it can work on the genome for genome targeting. The next thing, and these are the basis how we design the vectors. So, for example, for the case nucleases expression, we need an RNA polymerase, which is basically RNA polymerase 2. And this is the promoter sequence, and we need a specific sequence which is called the 5' UTR. And the transcription start from here, but the protein start from here. So this sequence just gave the stability to the protein. Specifically for big protein, these sequences are important. And then we have the coding sequence. Then there is a stop codon, and after stop codon, the protein, the amino acid sequence is stopped, but here we have 3' UTR region. And this is really important for the stability of this much big protein, specifically like Cas9 or Cas12, they are very big RNAs, and these sequences are basically provide the protection. And then the transcription stop site, which is normally, we call it terminators. In our experience, the promoter is important, but for the Cas9 specifically, the terminator is also much more important than the other proteins. The next one is the gRNA. So here, as we see, that in the gRNA case, the protein case, the Cas9 protein case, we need a specific sequence which is not part of the Cas9, but just for the protection of this express sequence. Here we don't need any extra sequence. Specifically at the 5' end of the gRNA, it's not tolerating more than 1 or 2 nucleotides. So if there is a long sequence, it will not work with the Cas9 and it cannot be used. The same is at the end. Even though the end is this 3' end is little bit flexible, but we should be careful, especially with the N nucleotides, that should be multiple T's, which gives stability to the gRNA structure. And when the gRNA structure is stable, it will be available for the Cas9. Then we have two different, in plants, we have two different systems, like we have dicots and we have monocots. So the basic thing is like when we are doing for dicots, we have to select the sgRNA expression, normally the RNA polymerase 3 promoters, which is normally a U6 promoter. and the Cas9 expression, which is the RNA polymerase II promoters. Normally, we are using Cas35S promoter, CAMV35S promoter, or ubiquitin promoter. In our experience, normally 35S promoter is not working that much better. It's better always to use a ubiquitin, which is not very constitutive, very highly expressed promoter, but the promoter or the protein produced from ubiquitin is very stable. Then for the monocot, we need the sgRNA expression. Again, we are using the RNA polymerase III promoters. Normally U3 promoter or U6 promoter. Compared to U6, here we are using only U6 promoter, but here compared to U6 promoter of the monocots, U3 promoters are working better than the U6 promoters. The Cas9 expression, for Cas9 expression, we can use these both. But the case, normally people don't want to use the 35S promoter in monocot, so that's why the ubiquitin and specifically the maize ubiquitin promoter is working very well. In the initial system that the CRISPR was developed, the first one was used as a 35S, like in protoplast, and specifically they use the protoplast from two different organisms, in Arabidopsis as well as in Nicotiana benthamiana. And they use 35S promoter to express the Cas9, and they put like the NLS and the flag tag just to detect these sequences, this protein produced. Also, they put an intron just to provide a stability into the Cas9 mRNA. And they put the NLS to take it to the nucleus, and then the NOS terminator. For the gRNA, it's the guide sequence, like the 20 nucleotide, and then the gRNA scaffold, and then the poly-T sequence, which gave a termination signal for the U6 promoter. So the difference between here, this promoter and this promoter, this is a termination signal which will keep it inside the nucleus. But here, the termination signal will take it from out of the nucleus, and it will go to the cytosol, and the protein will be made, and then so different RNA binding protein will bind to this transcript, and different single-stranded RNA binding protein will bind to keep it inside the nucleus. And this is the basic structure of the produced material. Like we have the Cas9, we have the gRNA guide sequence, which is complementary to the target sequence, and then we have the scaffold, which is responsible for the activation of the Cas9. So Cas9 have two types of activations. The first one is coming from binding with the scaffold, and the second one is coming from the Pam sequence. So when the Pam sequence is there, when the scaffold is there, then the Cas9 is activated. But then if this sequence match perfectly, it will make a cut. If not, it will go and it will scan for the right target. And then it was confirmed that how these systems are working in the two different organisms. So they use in Arabidopsis like ATPDS. They use two targets, ATPDS and FLS2. So here you can see that the system show only 5% of the editing, and here just 1%. But when the same system is used in the tobacco system, here you can see that the efficiency is very high in both targets. This means that we should be very careful about designing the vectors for the CRISPR-Cas system. And most of the people initially, most of the people try in Arabidopsis and they failed, and nobody get a real editing efficiency, a high efficiency in the Arabidopsis. And the reason is not only the promoter, but also the terminator they were using in the initial experiment for Arabidopsis. To improve this, different groups then start that why it's not working in Arabidopsis. So then like they select the X cell specific promoter, like to express the Cas9, and a specific terminator, that is the Robisco terminator from the peas, and then these two together, and the Cas9 was ZMS codon optimized, the plant codon optimized Cas9, and this system worked efficiently in Arabidopsis, and they use two different U6 promoter, so U626 promoter and U629 promoter to express the guide sequence for two different genes, the T and C, and ETC2. So this system confirmed that yes, it can work in Arabidopsis if you change the promoter and terminator. But again, the efficiency was very low. So our group also tried to enhance this system, and we used the meiotic specific promoter. So this promoter is expressed only in the meiosis. And we were thinking that if you can target the genome at the meiosis in the next generation, you can get directly the mutated plants without going to the T2 and T3 generation. So this system worked very well. And here we can see that compared to the original one, which is only 5% mutation, the meiotic promoter, we use 3 different meiotic promoters. The efficiency enhanced very robustly, and here you can see we can reach up to 33% just in the T1 generation. So this system is working very well in Arabidopsis, and also this can be adopted for every plant, specifically if we know the meiotic promoter or the XL specific promoter, and we can express the Cas9 at that stage. Then another system like this also opened the field that you can just target only genes that are expressed in the flowering, that in fluorescence, or shoots, or leaves, or in roots, or even the specific cells. Like in the leaf, you can just target specific cells, and only those cells genome will be targeted. And here you can see that when specific promoter are used, the Cas9 is expressed only in specific cells, not in other cells. The same, like for example, the ubiquitin is used as a control. So here you can see the whole plant is mutated. But when another promoter is used, the genes are only targeted on those tissues, but not as a whole plant. So this means that in the plants, we can target specific tissue or specific cells. Another interesting system that was recently developed was to avoid tissue culturing. Like this is a very good system. And here what they did, like for example, they use 2 different genes, the BBM and the WOSHL, and they express this from a binary vector, including Cas9. So they just inject infiltrate the agrobacterium expressing the BBM1 and WS. And then the regeneration starts directly from the leaves. So no need of tissue culturing of the hormonal balance or these things. You can just cut it and put it in the rooting media, and this system can work efficiently. So this vector system specifically developed to avoid tissue culturing and to do efficient genome editing. Then the next step was the binary vector for the monocots. So in monocots, as I explained, that the ubiquitin is working very properly for the Cas9 and the T-NOS terminator, and you can use the U3 or the U6 promoter to express the gRNAs. But this, normally in the cereals, we need multiplexing, and for multiplexing, a very good system was developed. That is the combination of the tRNA structure with the gRNA. So what they did, actually, they put a tRNA and then a gRNA structure, then again a tRNA and gRNA. So what happens that when this transcript is made, the gRNA, multiple gRNAs will be produced because of the two nucleases, that is the RNase-P and RNase-Z. So RNase-Z make a cut here and RNase-P make a cut here. And the gRNA will be released, and the multiplex system can be used very well. And this is one of the most widely used systems in the monocot system, including rice, barley, wheat. And this is working very well for the monocot. So to improve this system, one is the multiplex targeting. The second one is normally we need the repair, not only the editing. So if we can do like the repair, so we design another system that if RNA can be used, like for example, we design a gRNA, and then we put a repair template that can be used for the DNA repair as a template or as a complementary strand, and then this repair can, if we put the repair here, it can be converted into the DNA, and then we put like the cis antisense and cis-sense system And also we put it in trans, like we put the tRNA, gRNA structure. So the gRNA is there and the tRNA is there, and then the repair template is there. So it will be cut here and the repair template will be available for the repair. This is not the DNA sequence, this is the RNA sequence. So this system worked very well. And then here you can see if you put the mutation here on the repair template of the RNA, it can move to the DNA sequence, and then you can produce the BS resistance, the drug resistance seeds. This is the wild type seeds, and here you can see these seeds are resistant to the drug. Another system that, because we need the gene targeting and repair, mostly like if a gene is not working or if we can put a specific function to a gene, this is important. For that, another vector system was developed that is like the Cas9 and the Vir2 fusion. So all the system is the same, except we put the Vir2. Vir2 is the agrobacterium protein, which is basically transforming tDNA, which is bound with the tDNA and transfer the tDNA from agrobacterium to the plant. So if you have this Vir2, it can bind with any repair template, and the Cas9 can make a cut, and the repair template will be there, and this can be used for the repair at that site. So like for example, we have a repair template and including the right border for the Vir2 binding. We have Cas9 and we have the gRNA. And then we use this system to make this complex, and this complex can guide the target with the Cas9, and the repair template will be bound with the Vir2, and the system can use for HDR, and we can get the homology directed repair. So in the next step, we look for these systems that it's working or not in the plants. And here you can see that when this system is, we put specific mutation, which is giving resistance to the herbicide. And here we can see that when this mutation is inserted into the genome of the plant, we can see the mutation is there, and then we can see here that these repairs, like we can enhance the HDR up to 20%, which is a good amount for the plant system. The next system that we just confirm the next progeny of these plants that yes, this mutation or this system, vector system, is working permanently, and we can get the mutation in the next generation. One of the best system till now that is working is the binary vector system. But in plants, multiple viruses can be used as vectors for genome editing specifically. So multiple viruses are available, like they are DNA viruses or RNA viruses, and they can be used as vector for the gRNA expression. So one of the most widely used system is the TRV system. It have two genomes, like RNA1 and RNA2. So what we did, we put the gRNA to be expressed from the RNA2, and then we just put to one leaf. We put this system into one leaf of the plant, and then the virus will move. This vector will move to the whole plant, and the whole plant cells will be mutated, including the germline cells. And when the seeds are there, you can get directly mutated plants without any tissue culturing. So this is a good system for the research purpose. And then you can see that we can get the leaf that is infiltrated as well as the leaf that is not infiltrated but systemic leaf from here. And we can get mutation there. And in the next generation, we can see that yes, the mutation is heritable and the system can be used. The only problem with this system was the low efficiency, like it was only 3% of the progeny were mutated. So another group, they really did a great job, and they add actually a mobile signal to the gRNA. The problem with the TRV is that it cannot easily move to the germline cells. So what they did, they put a mobile signal to the gRNA, and now they can, they use our system, but they put just a signal, mobile signal to the gRNA in this vector, and then it can move to the germline cell. They basically put the FT locus signal, which is produced basically in roots and move to the shoots. So it's a 35 nucleotide sequence, which can move from one part to another part in the plants. And then they use this system, and here you can see that actually when you infiltrate this vector system into this leaf, they will move, and here you can see the plants are becoming, they are targeting PDS gene. So the plants, this part is becoming white, and here you can see that the PDS targeting is there. And this is in the next generation. So this FT locus is basically giving the chance to the RNA to move to these cells. And this is a good enhancement to our system. And the next, the same system was developed using a different virus, which is the monocot virus, the barley strep mosaic virus. And the barley strep mosaic virus, it have three-partite genome. It have 3 parts. So what they did, they just put the gRNA, and also they include the tRNA, because the tRNA, gRNA structure working very well in the monocots. And also they put the FT signal. So they can get easily, like for example, this is the genome, and they put the gRNA, and this is injected to one part of the plants, and then it's inoculated. It's first replicated in the tobacco, then put to the plants. And as this is RNA, there is no DNA sequence. So they just put the RNA to this one, and then you can get the mutated wheat in the next generation. And these are non-transgenic because there is no involvement of the DNA. This system is this vector system also works very well. But the most interesting one is the recently published one, where they put all the things, including the Cas9 as well as the tRNA, gRNA structure, into the virus, to the SYNV virus, and they express all the things from the virus. And this is RNA virus. So if we have this virus and this system, this vector system, we can use this one for the non-transgenic genetically modified plants, which is a very big concern. So what they did actually, if you have a plant, you just infect and you clone the Cas9 and gRNA, you just put this virus into the plant, and then you just get because this is RNA virus, it will replicate, and then you can get the new plants from these plants. So this system, or this vector system, is recently published, and I think this have a good future. And the other one, all these what I was presenting, this was about the RNA vectors. Then the DNA vectors can be also used because the DNA viruses normally they go to the plant nucleus, and there is a good chance that these can be used to express the Cas9 as well as the gRNAs. So for the monocot, they produce you just need the LIR sequences from these viruses, and then you can put the Cas9 material or the CRISPR-Cas system into this system. And when it's replicate inside the plant cell, you will see a lot of viruses produced compared to the single agrobacterium transformation. Here you have a huge amount of the Cas9 replicons. And the rate of the mutation is very high. Recently, another system, basically the same concept, but another system from the wheat dwarf virus was developed for monocot, where they use the ubiquitin promoter with the Cas9 and the U6 gRNA for the Cas9. But compared to the ubiquitin one, the normal Cas9 one, maybe this system is not tolerated by the virus. And here you can see that without the ubiquitin, the efficiency is high, but with the ubiquitin, the efficiency is low. Ubiquitin is not a viral promoter, and maybe it's not working as efficient as the virus promoters are working in the replicons. And here you can see that the system is working very well for these system. Recently, we are thinking about a virus system, and we are developing this system. So like, for example, you have the CRISPR-Cas system in the plant. You just cut the plant here. You inoculate the plants with this vector here. And then the next shoot will come out because the plants have good regeneration rate, and the progenies will be mutated. So we try this system, and we are working on it. So here you can see that when you inject the Cas9 GP, we can see that the plants become GP, the GP luminescence is here. and here and here, and this means that the new shoot coming out will be the new seedlings or the new shoot coming out will be expressing Cas9, and hopefully they will be mutated. We just did some sequencing and we can see that yes, some of these progenies, they are mutated. So if this is working, then it will be really good vectors for the without tissue culturing and no need for the transformation. Again, another problem in the plant system is the transgenics. So nobody likes the transgenic plants. The Cas9 can be translocated from agrobacterium because agrobacterium have the capacity to translocate its protein, virulence protein, to the plants. So it's a natural engineer. And we think that if we can translocate the Cas9, if we express the Cas9 in agrobacterium, putting the VERF signal with the Cas9, then this protein will be produced here, and then due to this signal, it will be translocated out of the agrobacterium. And if it can go out of the, and specifically if we produce the sgRNA also, so this complex is produced in agrobacterium, and this is as a complex, not any DNA or anything else, like no RNA, no DNA, only RNA and protein, and then it's translocated into the plant cell. And then it will go to the nucleus because it has the nuclear localization signal in the Cas9, and then it will mutate. So if this can work, the plants will be completely non-transgenic because there is no transfer of any DNA. We check this system, it's working very well in agrobacterium. And then we try it in plant system. And here we can see that up to 9% activity we can see for genome editing. But to enhance this one, we couple this system with the TRV system. So the Cas9 is coming from the agrobacterium, and the gRNA is coming from the TRV system that we already established and that I present in one of the slides. So the RNA is coming, the gRNA is coming from here, and the protein is coming from here. And then the whole complex is made and the genome is mutated, and here we can go up to 18% of the mutation. And here we can see that the next generation is mutated. Then the CRISPR-Cas-based vector can be used for virus interference because most of the viruses, they are going to the nucleus. And if we direct the Cas9 into the nucleus, we can target the viruses because the viruses are replicating in the nucleus. And if we produce the same complex inside the nucleus, so we can target the virus and we can cut the genome of the virus. So for that one, we just try first the TYLCV and the tobacco leaf curl virus as well as the cotton leaf curl virus. But here you can see that mainly the IR region targeting, the coat protein targeting, and the RCR2 targeting. You just need to express the gRNAs, and then you can see that the virus is mutated in these regions. And there is no accumulation of the virus anymore in these plants compared to the control plants. And you can see that when you are expressing the gRNA against the viruses, you can recover these plants. So the virus vectors can be used to provide resistance against pathogens, specifically viruses, because their genome is directly available inside the nucleus. We also use this system with the Cas13 to provide resistance against RNA viruses. And the Cas13 can process the gRNAs, so you can produce multiple gRNAs against the RNA viruses, and then it can target the virus. And there is no accumulation compared to the wild type, so the virus is not accumulating anymore in these plants. And here you can see the plants are resistance. The plants are providing very good resistance against these viruses when we are expressing these system. So in conclusion, the vector selection is the core for efficient plant genome editing. An expression of the CRISPR-Cas system in monocot and dicot have specific requirements. You cannot use the monocot one in dicot or the dicot in monocot. They have specific requirements and they need it. The multiplex targeting is possible. Cas12 by itself, it's processing its gRNA. But for the Cas9 targeting, multiplex targeting, we need the tRNA, gRNA, or the ribozyme. Both DNA and RNA viruses vectors are available, and the deconstructed virus can be used. They are very efficient compared to the normal binary vector system or the translocation system or to the RNP system. Tissue-specific or phase-specific vectors can enhance the recovery of the genome edited in next progeny. And RNA vectors and genome editing reagent translocation can provide non-transgenic editing of the genome. This is all about my presentation. This is our group. This is our group leader, Magdi Mahfouz. And this is Dr. Haroon working with rice, basically. This is Rashid, he is working with the Dicots. Khalid also joined recently, and he is also working with rice. The rest of the group, they are the PhD students, and they are really brilliant students, especially Ahmad. He is working now on the diagnostic using the CRISPR-Cas system. Thanks for your attention, and if there is any question.
Okay, thank you very much for this great presentation. Now, do you have any time to continue with us in this presentation, or should we take the questions for you, Zahira?
It's okay. It's okay. I can meet here.
Okay, if you can just stay for Mark's presentation, then we can take Mark's presentation right now, and at the end we can have a general discussion altogether.
Okay, okay. Yep.
Okay, thank you so much. We appreciate your presentation. Now we have our final speaker, Mark Smedley, coming from UK. And now first I need to take his presentation. Give me just 1 second. Ankara yazma. Ekranda paylaşmışsınız.
Nereye paylaşsam? Açıldı mı söz?
Açıldı onu.
The pointer da açarsan sevinirim.
Tamam. Orası normal. Burada sadece böyle. Pointer burada değildi ama oraya şöyle. Biraz durulabilir.
Nasıl olacak mı?
Şuradan. Oraya bakıp buradan.
Tamam. Bak.
Yok değil galiba. Tamam oluyor.
Saatini değiştireceğiz. Tamam mı? Tamam.
You can sit it here. Brief information. Now, basically, now our next speaker is Mark Smedley. He's joining us from our Genome Chapter. He has been transgenic plants for a very long time. We are really good at wheat and barley transformation. So today he will talk about us. He will give us a brief example, like a case study on RNA design and philosophy.
Thank you. Can everybody hear me? So, I'm going to give a on edits in with
Yes. Okay. I think now it'll work. Okay. That's it.
Okay. Take. So this complex wheat genome came about by two wild species, two wild grass species, crossed about 400 years ago. This gave us the wild Emina wheat. And then about 10,000 years ago, it crossed with Taoxiai to give us our bread wheat, or was domesticated and became our bread wheat. And likewise, the wild Emina was domesticated and became our pasta wheat. So this all actually happened southeast of Ankara in Turkey. And this sort of played out like this. So the wild Emina cross was as I said, southeast in Turkey. There was two domestications, one here in Turkey, and one the southern domestication. And then moving over further to what is now Iran, the crossing with Taoxiai came about, and this was then domesticated in this area. So this complex genome means that we've actually got 3 copies of every gene, and there's 97% similarity, which can cause problems, but can also make targeting all 3 easy sometimes when designing guides. Similar expression patterns mostly across the 3 subgenomes. There's huge plasticity, and it's a dynamic. And there's a buffering effect, which for genome editing, if you're doing knockout, then invariably you want to knock out all 3 genomes. So we actually see with some genes that we actually get an additive effect. So if we knock out A, we will get a knockdown. If we knock out B, you know, we will get maybe a third less expression, but we need to knock out all 3 genomes to completely knock out a gene. So we've already designed this wheat. We're using 2 whole 3 promoters. We're using the wheat, the TA use promoter or TA promoter, and our guide RNA is cloned downstream of that. You can see the scaffolds in here in the blue. Is where the Cas actually locks into the guide RNA. And for transcription start, we generally start with the U6. We start with a G in our guide, and with a U3, we generally start with an A. And if in the guide sequence there isn't actually a G, we can add an additional G. So designing guides, guide RNAs in wheat. So if you want to target a single copy of a gene, and we have done that in the past. I think our first publication in wheat was targeting just one single copy. and I think Shadia will talk about that tomorrow. The best way or the easiest way that I find to do it is to actually do an alignment of all three genes, looking at the coding sequence, and I tend to design two guide RNAs I tend to design 4 guide RNAs and clone 2 guide RNAs in per construct, and 2 constructs per target. So when designing a guide RNA for to knock out a single copy of a gene in wheat, I will line the sequence, and then I will look for differences. I will look for differences in the sequence, and I will then design the guides in around those differences. However, if we're planning on knocking out all 3 subgenomes, then I tend to align the sequence, and I then look for similarities. I look for areas of homology. I can then design my guides, and these guides will target the guides to the right of the screen. You can actually target all three copies. So I would then choose this is a CRISPR Wheat. This is an online tool. The nice thing about this is that it's actually linked to the wheat genome, which is the Chinese spring genome, and which means that it look for off targets. It can give an off target score as well as an on target score. And there is a tick box when you put in your sequence. There's a tick box which basically says, do you want to target just that one gene, that one subgenome, or do you want to target all 3 copies of the box? Will tell you the guides to target all 3 copies of the gene. Now, I would like to say that generally don't work with Chinese spring. So invariably, what we used to do was actually PCR through the gene of interest in our target species, which was Fielder, which was the transformable, then design our guides on that sequence, or check our guides on that sequence to make sure that it's okay. Charlie is going to talk tomorrow about a number of different wheat species that we're transforming and genome editing in, and we now have the sequence, the genome sequence of Fielder, Cadenza, Kronos, Paragon, and several other wheat species as well. So you know, it's getting easier. All tools are. So now we've sort of designed our guides. We then need to think about cloning strategies. And we're using a Golden Gate system for ours, and I'll just give an overview, and then I'll show you some of the wheat vectors specifically designed that we're using. So Golden Gate cloning. Yeah. Just go back. I'll just have to talk to you. Okay. So Golden Gate cloning relies on these type 2S restriction enzymes. Now the beauty of these are that they have a recognition sequence, and then they miss a base, and then they cut, leaving a full nucleotide overhang. Unlike sort of traditional cloning, you can use one or two of these type 2S restriction enzymes, but you can have a multitude of overhangs, and you can choose those overhangs as you go. So Golden Gate cloning basically is a one-pot cloning method, and you would have your promoter, your coding region, your terminator. You would put this together with your type 2S restriction enzyme. In this case, we're using BSA1, ligase, and a acceptor plasmid, such as this Golden Gate vector here, which has the LAC-Z for blue and white screening. The tube then goes into basically a PCR machine. It goes through rounds of 37 and 16, so cutting the DNA, ligating the DNA. Because the components are actually cut out of these first plasmids, leaving the with these overhangs, and leaving the enzyme recognition sequence within the vector backbone. Once these have ligated and they're stuck, they are stuck kind of permanently. The enzyme won't come again. So if you then put another restriction enzyme together, in this case I've put BPI, this can then take out the cassette that you've just built and clone into another acceptor vector. And by using this method, you can actually make very complex vectors very, very quickly, and very easy. It's a very robust system. So we're actually using what we call the mole-close system, modular cloning system. It has set overhangs, and it's hierarchical in its system. So the level ones would be your individual promoters, coding regions, terminators. Your level twos are your transcription cassettes, sort of put together already in these into these level ones. These each have a position. So the overhang, when they're cut out with BPI, their overhangs will align them into a level 2 acceptor, and this will be your final finished vector. And you need a linker here which links the very last component to the acceptor, to the right border of the acceptor. As I said, we're using the mole-close system, and this has a number of set overhangs, which means that you can easily put together your promoters, your coding regions, terminators in one pot reaction. So with wheat and cloning your guide RNAs, we're doing a very similar system to the speaker, in that we order 2 complementary nucleotides guide RNA sequence, and we have our are in red here. We then do a Golden Gate reaction, and these are then dropped into level 1 position. And I will explain a little bit in. Removing and it's very efficient percent white colonies. Once your level 1's made, you've got your guiders, and then you need to put together your level 2 vector or your binary vector, which the positions on these Golden Gate vectors is basically position from the left border going across the right border, which is position 7. And as you can see, for wheat, we will have our selection. We always put the selection gene close to the left border. That's basically because the tDNA goes in via the right border, and you can get truncation at the left. So obviously, if your selection goes there and you get truncated selection, you won't select those plants coming through. We were actually using the rice actin promoter driving the hygrorhizin selection for wheat, but we have started using the switchgrass UB2 driving the high selection as well. To drive the Cas9, we're actually using the rice ubiquitin promoter. We find that a little bit stronger than the maize ubiquitin promoter, and we're getting better results. And in position 3, position 4, this is where we've previously cloned our guide. And of course, you have a linker here. These all go together in a one-pot reaction vector. This is our level 2 PGG or P Golden Green Gate. This is a vector that I made. It's P green based, but I've put an additional left board in it, and I've got Golden Gate compatible. And all the vectors are actually available on AdGene at the John Innes Centre Crop Transformation and Genome Editing page. Your final vector will look like this, and it's just then a case of sequencing, putting into agrobacterium, and then transforming into wheat. So, as I said, Dr. Shadi Hayter is going to be talking about the wheat transformation tomorrow, and we'll show some of the genome editing results that we've actually got. The vectors and the transformation protocol are both together in current protocols, and it's open access as well for those who want to go into more detail. So once you've transformed your plants, you're getting little plantlets back. You then need to start to think about screening. Hopefully, you might have actually thought about screening before that, but it's not always the case. With wheat, because of the complex genome, there's several ways that you can do it. The easiest way that we find is to actually design genome-specific primers that will amplify through each copy of the gene, and we just directly sang a sequence for across the target area. We also run a little bit out on a gel because we're using a 2-guide system. If the guides have worked simultaneously and you've got a large deletion, you'll usually see that on a gel, and it's a nice way of actually following edits in the next generation, in the T1 generation. So once you've got your sequence back, you can look at your sequence trace. And this is a typical kind of edit that we would see. So here's your guide RNA, here's your PAM. You expect Cas9 to cut between the third and the fourth base from the PAM motif. So you'd expect to cut here. And it's just a case of looking through the traces and looking where the guide is, and you see this this drop in signal, and again these kind of double peaks, and this is basically where two products are then going through the sequencer, and the sequencer doesn't know which bases to call. At the moment, we are using next generation sequencing to look for edits, and we're finding this very accurate. But with Illumina sequencing, the products are very small, they're about 150 bases. And if you get a large deletion, it can be problematic trying to identify those with next generation sequencing. We're now looking at nanopore and setting up in-house nanopore sequencing, which will give us the best of two worlds, if you like, in the sense that nanopore will be able to sequence a 500 base pair product. And you will have the accuracy of next generation sequencing. So, so I've basically talked through here with knockouts. This is kind of the basic knockout system that we're using. It's most popular, but we're also doing so as and I'd just like to finish up now just by thanking the people or recognizing people in our group, particularly Shadia for the wheat transformation, which is certainly the best that it's ever been at John Minutes. I'd also like to thank Martha Clark, Shadia's RA, who helps me quite substantially with the cloning, and also Maria and Macarena for all their plant work. I'd also like to recognise some of our collaborators in Christobal's group, in Graham Moore's group, and Jorge and Juan in UC Davis. And thank you very much.
Thank you very much for the presentation. Now we can take the questions for all three. Any questions from the digital participants?
Hi.
Thank you for your presentation. So actually, I can ask you also like you run also. So did you try any root specific for the expression of time? Because probably people now using yes, promote maybe, but like any good promoter for root specific or any other form specific promoter. Did you try that one?
No, we haven't. I mean, so if would you just want to produce like a chimeric plant? So producing your in the roots, maybe knocking out a gene in the roots, but not knocking it out in the top part of the plant, or
my aim is to overexpress some genes in just food. So
right.
in CRISPR system, so just for example, I will overexpress some gene in just root. So I'm going to use root specific promoter in case time.
Okay.
In literature, so I don't see any strong root specific promoter. So
I don't actually know of any strong root specific promoters. would probably be somebody who's working on sort of bacteria and fungi with roots would probably know much more than me on root specific. But you can always enhance promoters or enhance your Cas9 with intron enhancement. I mean, it's been shown in Arabidopsis that they put 13 different introns in the Cas9 and expression was much higher. I do know that a lot of weaker promoters in wheat and in barley, we've actually used the leader intron from the ubiquitin promoter, the maize ubiquitin promoter. It's 2 kb long, but 1 kb of that is actually a leader intron, and by positioning that downstream of a wheat promoter, that actually enhances the expression of a wheat promoter.
Seed cotton, reduce the cost of phosphorus in the cotton seed so that it can be utilized by trees. So even down that so the seed stuff. So the expression of Cas9 is required for the cotton on the other part to give protection against it. So we have done this. What is efficiency? So efficiency was because it was expressing at the same the level of expression of phosphorus in the leaves, and the upper part was the same part in the seeds.
In order to elaborate this question, can we ask exactly the same question to Zahir Ali as well? Do you know any promoters that are specific to the roots that can be used?
I don't know exactly, like because we are mainly working with the progenies, so I'm not working with the development. But definitely, like for example, Ikram, she is one of the leading scientists in developmental biology. So I think you can look at her work, and definitely she did some work with the root promoters, specifically with the auxins promoter. I think the auxins promoter, producing promoters, they are basically root specific promoters.
Okay, now we can take one more question.
So I have a question for Zahir. Please available. Zahir?
Yep.
Can you hear me? So my question is that, what do you suggest, the system for vegetative crop like potato and cassava and banana? So, what could be the ideal system for every genome level? Because, there's no seed system, so obviously we don't need to remove the pests. What is the solution?
Can you repeat the question? I didn't got the exact.
This is about vegetatively grown crop like potato, cassava, banana.
Can somebody repeat near to the mic? I cannot hear the voice.
Sorry, I have to compare.
Okay, Zahir, my question is about genome editing in vegetatively grown crops, so like potato or cassava or banana. So what system you suggest? Because in case of crop that where we have a system available.
Yep. Actually, I present some of the. I will just look at that. back. I think like the translocation system is working better for the especially vegetative one, because we cannot remove the transgene from there. So the agrobacterium mediated translocation system is working much better for the vegetative system. So I think if this can be applied like in potato or vegetative crops, I think it can be used very well.
And one more question to Mark is about cis-acting elements to actually not to knock out, but change the expression levels.
That's also possible.
Yeah, sure.
So maybe I can answer both questions actually. So the what we're actually doing with wheat in one particular case is that we've got a heat shock promoter driving a Cre recombinase, which has an intron in it, and we just put lots of P sites either side of the tDNA, and then heat shock the material when we want to remove the tDNA, and the tDNA is recombined out. It does leave about 16 bases, I think. So it's not absolutely clean, but that's almost as good as clean. I mean, 16 bases is less than an oligo. Sorry, could you repeat your question again? Yes, we are at the moment actually. So we're actually targeting promoters, and we've designed. We're multiplexing, and we've designed 4 and 6 guide RNAs for several different promoters, and we're hoping that the guides won't be very efficient, so we'll get a lot of variety in those promoters coming through. So obviously, if the guides were very efficient, and we're targeting maybe 2 kb of the promoter region, if they were very efficient, then they would just take out 2 kb of the promoter, and and that would be it basically. But what we want is to target. We're targeting different kind of sites within that promoter, hopefully to change the expression levels either up, down, or in time, or spatially or spatially.
Any other questions from the audience? More general question.
This one works better.
Yes. I don't know whom to turn my back. So more general question. If you number the difficulties working with wheat, regarding CRISPRing wheat, what would be the most difficult, the biggest challenges working with wheat while doing the CRISPR compared to Arabidopsis or rice, for example?
So I think actually targeting just a single gene within one genome, that can often be quite problematic. Certainly with 97% homology, actually choosing guides is very, very difficult when you're doing that. But again, the tools have increased, so we're trying different Cases and different Cas proteins within wheat. They have different PAMs. So if one PAM is sort of available in one particular genome but not in another subgenome, then that will obviously make it a lot easier. So that's quite a difficult thing. The other thing is that with crops, they tend to be genotype dependent. So you can only transform perhaps one type of one variety, if you like. So with wheat, it was always Bob White that people would transform, and then Fielder came along and people started to transform Field because it was more efficient. And Kronos transformation is fairly inefficient, but that's a tetraploid wheat, a pasta wheat. Well, there's a lot of resources in there. And now we have a technology, a GRF technology, Charlie's going to talk about tomorrow, that has actually broken down all those barriers. So her transformation efficiency for a tetraploid wheat now is about 80%, and the same for many of the other wheat varieties. We're getting high transformation efficiency. And obviously, if you can transform different varieties and produce a lot of transgenic plants, then it's easy to try and, you're just sport for choice for actually looking for those edits, and if you're looking for knockouts, you then just look for the large deletions. You don't bother with the small ones. So yeah.
Okay, are there any questions? Are there any questions from the physical audience? Can you come to the stage to ask that?
Hi, Mark. I have a question. When you are using a modular cloning system, how do you choose the promoters and terminators?
How do I
choose promoters and terminators when you are using modular cloning system?
Can you repeat the question?
Yes. How do I choose when I'm using a modular cloning system? How do I choose promoters and terminators? It really depends on the species that you work in. So if I was working in, I would use promoters that are dicot promoters that are specific for those. We actually find that the cassava mosaic virus terminates across the cereals, across the 35S terminator or the NOS terminator, and they work quite well in all the different species. But it's not so much choosing the promoters or the terminators because of the modular system. It's choosing them because of the plant variety that you're actually working in. So in Brassica, for the pole 3, we're actually using the U3, the U6 26 to drive the guides. We find that works very well. So it's just really down to the species that you're working in. Most of the things that work in wheat will work in barley, but in barley selection, a very strong cereal promoter driving the selection, the hygromycin. We'll just have the 35S driving the hygromycin, but that is intron enhanced with the Cat1 intron.
Now, I think we can take a few questions from the online. Okay, actually, this chat is also recorded. I think we can share that with you. Now, let me just 1 second. Basically, Dr. Hansel mentioned about 2 papers about some root experiments in rice. So when we share those box things, so you can just see those comments. Now let me see if there are any other questions. Yeah, I think there are no other questions from the online audience. Now I have one other thing related to the root-specific promoters. I think there was a paper published from Ben Fee's group a long time ago, I think in 2008. At that time, they generated some RFP constructs, and those constructs actually are defining the promoters were from different parts of the root tissue, and they were just comparing their activities. So maybe if you can find that paper, that can help you. Okay, we have one question. Are there any chances of allergy development during wheat transformation? Any comments?
I think that should be addressed to actually. I do know somebody who worked with barley for quite a long time and ended up getting allergic to barley. And imagine that if you do work with one particular, perhaps likely to get censored, kind of answers the
So we came to the end of the today's lectures. I really thank all the participants, all the lecturers. Thank you very much for your efforts. Tomorrow again we will start on at 9 o'clock Istanbul time, and then we will continue the rest of the lectures. Thank you very much.